As you rest and prepare to drift gently into sleep, imagine the quiet unfolding of life's deepest foundations. All around you, within your body and beyond it, the chemistry of life is at work. It moves silently, invisibly, without demand for attention. In the rise and fall of your breath, in the warmth of your skin, in the soft beating of your heart, there is a hidden choreography, a choreography of molecules, of atoms, of reactions so small and so quiet that they are often overlooked. Yet it is in this subtle, constant dance that life itself is sustained. This is the world of biochemistry. It is, in essence, the study of life at the molecular scale, where chemistry, the science of matter and its transformations, meets biology, the science of living things. There exists a bridge. That bridge is biochemistry, connecting the invisible bonds of atoms with the visible growth of trees, the flow of rivers, and the existence of you. To begin, let us ease into this world with a simple image. Imagine holding a droplet of water on the tip of your finger. To the naked eye, it is nothing more than a bead of clear liquid, fragile and small. Yet within that single droplet exists a universe of interactions. Countless molecules of water link and unlink through tiny bonds. If nutrients are dissolved within, they drift softly between the molecules, carried by currents invisible to us. If it were a droplet from within a living cell, proteins would be folding into shape, sugars would be breaking apart to release energy, and strands of nucleic acids would be carrying instructions forward in time. All of this occurring silently, tirelessly, without pause. When we use the word biochemistry, we are not speaking of something harsh or mechanical. Rather, we are speaking of something delicate and balanced. Every living process, from the way leaves capture sunlight, to the way our muscles contract, to the way memories are formed, depends on a quiet set of chemical principles. These principles never rest, but they are not rushed. They operate with steady patience, much like the gentle flow of a stream that carves valleys over ages. Biochemistry is built on a few simple truths. The first is that life depends on a handful of key elements. Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur form the majority of living matter. These elements are not rare, nor are they exotic. They are common, scattered across the earth, the oceans, and the air. Yet when arranged in certain patterns, they become the molecules of life. They form sugars that nourish, lipids that insulate, proteins that build and repair, and nucleic acids that record and transmit information. The second truth is that life depends on water. Nearly every biochemical process takes place in water, the universal solvent. Its ability to form bonds, to dissolve salts, to flow freely yet hold together, makes it indispensable. Without water, there is no biochemistry as we know it. This is why, when scientists search for life on other planets, they begin by asking a simple question: is there water? The third truth is that life requires balance. The chemical reactions inside a cell do not occur randomly or chaotically. They are regulated, adjusted, and coordinated in such a way that conditions remain stable. This stability is called homeostasis, and it allows organisms to survive even as their external environment changes. Think of your own body maintaining its temperature, your blood sugar, and your hydration. These balances are maintained by chemistry, soft, unceasing reactions that restore equilibrium whenever it drifts. Biochemistry can seem at first like a collection of details, structures of molecules, names of pathways, steps of reactions. But if you listen quietly to the story, a pattern emerges. It is a story of cooperation, of molecules working together, of processes linked in delicate chains. Each reaction supports the next. Each molecule finds its place in the greater whole. Nothing is truly isolated. In this way, biochemistry reflects life itself, interconnected, balanced, and continuous. Perhaps the most comforting thought is that the same chemistry of life flows through all living beings. The glucose that fuels your brain tonight may have originated in the leaves of a tree, drawn from sunlight and carbon dioxide. The oxygen filling your lungs may have been released by algae floating in the sea. The calcium in your bones once rested in stone, weathered by rain, carried into soil, and drawn up by plants. When you look closely enough, you find that every atom in your body has been part of the earth for billions of years. You are not separate from nature, you are made of it. As you drift deeper into this gentle reflection, consider the continuity of life. For over 3 billion years, molecules have been combining, separating, and recombining, from the earliest cells in ancient oceans, to the great forests, to the quiet beating of your own heart. The chemistry of life has remained. The forms have changed. The patterns have grown more complex, but the principles endure. A bond between carbon atoms, a hydrogen atom joining with oxygen, an electron shifting from one molecule to another. These same events have been happening since life first began, and they continue within you now. Biochemistry is not only a science of molecules, it is also a science of energy. To live is to require energy, and chemistry provides the means of obtaining and storing it. Plants capture sunlight and turn it into sugars. Animals eat those sugars and break them down to release energy. Microbes recycle matter back into simple forms. In every case, the molecules change, but the energy flows onward, never lost, only transformed. This too is a source of calm reflection, that life is not wasteful. Every reaction is part of a greater cycle, a cycle that has been turning for ages beyond measure. As we move through this lecture in the hours ahead, we will explore each of these ideas more closely. We will pause with the atoms, the smallest building blocks of matter. We will spend time with water, uncovering its quiet but extraordinary traits. We will follow carbon as it forms the backbone of molecules, weaving endless patterns. We will examine the 4 great families of biomolecules: carbohydrates, lipids, proteins, and nucleic acids, and see how together they sustain the body and the natural world. We will watch how energy is carried in molecules like ATP, how it is released in respiration, and how it is captured in photosynthesis. And finally, we will reflect on how these processes, though small, connect all life on Earth into a single fabric. For now, though, there is no need to hurry. Rest with the knowledge that biochemistry is the quiet heartbeat of existence. It is not loud nor demanding. It is not separate from you, but within you. With every breath, with every gentle beat of your heart, molecules continue their patient work. They do not ask for awareness, yet, in giving them your attention for a moment, you may find a sense of calm. The chemistry of life is not only a subject to study, it is a reminder that even in silence, even in stillness, there is motion, there is balance, and there is continuity. And so, as you drift toward sleep, let this thought stay with you. The same molecules that sustain you tonight will sustain you tomorrow, just as they have sustained life for billions of years. You are carried by them, supported by them, and connected through them to all living beings. This is the gentle promise of biochemistry that life, in all its forms, is one unbroken current. 2. The building blocks of matter. As we continue our quiet journey into the chemistry of life, let us now drift gently into the most fundamental layer of all, the building blocks of matter. Before we can understand cells or the molecules that sustain them, we must pause and reflect on the smallest units of existence, the atoms and the elements from which all life is constructed. Picture yourself walking through a forest in the early morning. Mist clings softly to the air, droplets glisten on leaves, and the ground beneath your feet is alive with unseen processes. The trees draw water upward, the soil nurtures roots, and within each cell of every living thing, the same small particles move about in silent cooperation. What you are seeing is vast and complex, yet beneath it all, at a level invisible to the eye, it is composed of the same handful of building blocks, arranged and rearranged in endless combinations. At the very foundation of this structure lies the atom. An atom is not a thing you can see directly, but you can imagine it as a small sphere, a delicate center, surrounded by even smaller particles in motion. At its heart is the nucleus, a dense core of protons and neutrons. Around this nucleus dance electrons, light and swift, tracing patterns of probability rather than fixed orbits. Though they are unimaginably tiny, atoms are abundant beyond measure. Every breath you take contains trillions upon trillions of them, passing quietly into your body, exchanging with your cells, and then flowing back outward into the atmosphere. Despite their simplicity, atoms are diverse. Each kind of atom belongs to a category called an element. Hydrogen, for example, is the simplest element, consisting of just one proton and one electron. Oxygen, so essential for life, contains 8 protons and a cloud of 8 electrons. Carbon, the great architect of organic chemistry, has 6 protons, 6 neutrons, and 6 electrons, capable of forming bonds in multiple directions. These basic variations give rise to the entire periodic table, a catalog of all the known elements in the universe. Though the table may seem vast, life itself relies primarily on a select few. More than 90 elements exist in nature, but only about 24 are commonly found in living organisms. Among them, carbon, hydrogen, oxygen, and nitrogen make up the overwhelming majority of matter in the human body and in most forms of life. Together, these 4 elements account for more than 95% of your mass. The rest, phosphorus, sulfur, calcium, potassium, sodium, and trace metals play crucial but more specialized roles. Life, in all its diversity, emerges from this modest toolkit of elements, reused and recombined endlessly. The bonds that form between atoms are what allow complexity to arise. Atoms are not static, they seek stability, and they find it by joining with one another. These connections are called chemical bonds, and they come in different forms. The strongest, covalent bonds occur when atoms share electrons, creating molecules with lasting stability. Ionic bonds form when one atom donates an electron to another, resulting in oppositely charged ions that attract each other. Hydrogen bonds, though weaker, provide the delicate glue that holds water together and stabilizes the structure of proteins and DNA. Without these bonds, atoms would remain isolated, unable to assemble into the larger molecules required for life. It is often said that life is built from molecules, but molecules themselves are simply collections of atoms bonded in specific ways. Consider water, perhaps the most important molecule of all. It is composed of just 3 atoms, 2 hydrogens, and 1 oxygen, yet its properties are unlike any other substance. Or think of glucose, a sugar that powers much of life. It is formed of carbon, hydrogen, and oxygen atoms arranged in a gentle ring. Proteins, the workers of the cell, are long chains of amino acids, each built from atoms arranged in repeating patterns. DNA, the carrier of genetic information, is a sequence of nucleotides, each one built from carbon, nitrogen, oxygen, phosphorus, and hydrogen. The endless variety of life's molecules comes not from countless elements, but from the endless rearrangement of a small handful. If you pause for a moment, you might find this thought comforting. The same atoms that make up your body have existed since the birth of the universe. They were formed in the interiors of stars long before the Earth was born. Hydrogen, the simplest atom, emerged in the first moments after the Big Bang, more than 13 billion years ago. Heavier atoms like carbon, oxygen, and iron were forged in the hearts of stars and scattered into space by stellar explosions. Over time, these atoms came together to form planets, oceans, and eventually the molecules of life. When you breathe in oxygen, or drink water, or eat food, you are taking in atoms that are older than the Earth itself. You are, in a very real sense, made of stardust. Within the human body, atoms arrange themselves into an exquisite hierarchy. Atoms bond to form small molecules like water, oxygen, gas, or carbon dioxide. These small molecules then combine into larger macromolecules, carbohydrates, lipids, proteins, and nucleic acids. Macromolecules form structures inside the cell, like membranes, organelles, and scaffolding. Cells join to create tissues, tissues combine to form organs, and organs integrate into the body as a whole. At each level, the foundation remains the same, the humble atom, quietly bonded into place. The stability of life depends on the way atoms interact. For instance, the balance of sodium and potassium ions allows nerve cells to send electrical signals. The arrangement of calcium ions enables muscles to contract and bones to strengthen. Iron atoms, nestled in the protein hemoglobin, allow oxygen to be carried through your blood. Even trace elements like zinc, copper, or iodine, though required in tiny amounts, are indispensable for enzymes and hormones. In this way, the chemistry of life is not just about abundance, but about precision. Every atom has its place. When you imagine the elements within you, it can be soothing to think of them not as separate or foreign, but as part of a cycle. The oxygen you breathe was once part of the atmosphere, released by plants during photosynthesis. The carbon in your cells may have been part of a leaf, a piece of fruit, or even a breath exhaled by another living being. The water in your blood once fell as rain, flowed in rivers, and cycled through clouds. Atoms pass continuously through living and non-living systems, never lost, only transformed. This continuity emphasizes a gentle truth. You are deeply connected to the world around you. The same elements that sustain you also sustain trees, birds, fish, and even the smallest microbes. The carbon in your DNA is no different from the carbon in the soil. The calcium in your bones is the same as that in seashells. The iron in your blood is the same as that in mountain stone. At the level of atoms, all life shares the same heritage. As we close this part of our journey, Let us reflect on the quiet permanence of these building blocks. Atoms do not age in the way organisms do. They do not tire or wither. They move from form to form, molecule to molecule, being endlessly reused. The atoms in your body tonight may one day return to the soil, to the sea, or to the air, and from there find their way into new forms of life. This recycling is not an ending but a continuation. It is part of the gentle rhythm that sustains all existence. And so, the building blocks of matter remind us of something profound, that complexity arises from simplicity, that diversity is born from a small set of repeating parts, and that life is woven from atoms as threads are woven into fabric. Quiet, patient, enduring. These are the true foundations of the chemistry of life. 3. Water, the universal solvent. As we move deeper into the chemistry of life, let us pause to rest with one of the most familiar, yet most extraordinary, substances in existence, water. It is everywhere around us in rivers and oceans, in the air we breathe, in the food we eat, and flowing within us, making up most of our very bodies. So commonplace that it is often overlooked. Water is at the same time utterly unique. Its quiet properties are what make life not only possible, but resilient, delicate, and continuous. Imagine a calm lake at dawn. The surface is still, reflecting the sky in shades of soft gray and pale blue. Beneath that stillness lies a hidden world of molecules, each one in constant motion, drifting, colliding, forming, and breaking bonds in endless cycles. This gentle motion is what gives water its character, a balance between cohesion and flexibility, order and fluidity. It is no exaggeration to say that without water's peculiar chemistry, the earth would be barren, silent, and lifeless. At the heart of water's nature is its structure. Each water molecule consists of 2 hydrogen atoms bonded to 1 oxygen atom. Yet the arrangement of these atoms is not in a straight line. Instead, the molecule has a bent shape, with the hydrogen atoms at an angle of about 104.5 degrees from one another. This seemingly small detail has vast consequences, for it means that water is a polar molecule. The oxygen atom, with its strong attraction for electrons, carries a slight negative charge, while the hydrogen atoms carry a slight positive charge. This separation of charge allows water molecules to form hydrogen bonds with one another. Imagine each molecule reaching out with tiny arms, holding gently onto its neighbors. These bonds are not permanent. They form and break constantly, in trillionths of a second. Yet together they create a network, a lattice of connections that gives water its remarkable properties. This subtle dance of attraction is what allows water to behave so differently from most other liquids. One of water's most important traits is its ability to dissolve substances. It is often called the universal solvent, because more materials dissolve in water than in any other liquid. When salt is sprinkled into water, the positive sodium ions are surrounded by the negative ends of water molecules, while the negative chloride ions are embraced by the positive hydrogens. The salt crystals break apart and disappear into the liquid, evenly distributed. Sugars, proteins, gases, and countless other molecules undergo the same process. Inside your own body, nutrients, minerals, and even oxygen are carried from cell to cell by this dissolving power of water. Because of this property, water acts as the medium for life's chemistry. Inside every cell, molecules float and interact within a watery environment called the cytoplasm. Enzymes meet their substrates here. DNA and RNA are transcribed and translated here. Ions flow in and out of membranes, carried by water. Without this solvent, the delicate reactions of life could not proceed. Another of water's quiet gifts is its cohesion. Hydrogen bonds cause water molecules to cling to one another, creating surface tension. This is why droplets form, why small insects can walk across a pond, and why water climbs upward through narrow tubes in plants. Cohesion also leads to adhesion, water's ability to stick to other surfaces. Together, these forces allow water to rise through the xylem of trees, carrying minerals and nutrients from roots to leaves. Without cohesion and adhesion, towering forests would wither, and even the smallest plants would struggle to live. Water also has an unusually high specific heat, meaning it can absorb or release large amounts of energy without experiencing drastic changes in temperature. This gives water a stabilizing role in the environment. Oceans and lakes moderate climate by absorbing heat in summer and releasing it in winter. Within your own body, the water in your tissues prevents dangerous swings in temperature, keeping cells comfortable and stable. This property is one reason life can endure through changing seasons, harsh weather, and daily fluctuations. Linked to this is water's high heat of vaporization. When water evaporates, it takes a great deal of energy with it. This is the principle behind sweating. As sweat evaporates from your skin, it carries away excess heat, cooling you gently. Plants, too, rely on this process, releasing water vapor through their leaves in a form of temperature regulation. This quiet exchange of energy between water and air contributes to the stability of entire ecosystems. Another remarkable feature is that water is less dense as a solid than as a liquid. Most substances grow denser as they freeze, but water does the opposite. As ice forms, its molecules arrange into a crystalline lattice held apart by hydrogen bonds. This makes ice lighter than liquid water, allowing it to float. This simple fact carries profound importance. When lakes and rivers freeze in winter, the ice forms a protective layer on top, insulating the water below. Fish and aquatic plants can survive beneath the frozen surface, sheltered until spring returns. Without this property, bodies of water would freeze from the bottom up, destroying aquatic life each year. Water also participates directly in chemical reactions. In hydrolysis, water breaks larger molecules into smaller ones, a vital step in digestion and metabolism. In dehydration synthesis, water is released as small molecules join into larger structures, forming proteins, nucleic acids, and other macromolecules. Thus, water is not only the stage on which biochemistry takes place, it is also an active participant in the performance. The presence of water has guided the course of evolution itself. The earliest life on Earth emerged in oceans, where water provided a protective shield against harsh sunlight and temperature extremes. Even today, most organisms maintain internal environments that mirror the chemistry of ancient seas rich in salts, buffered by water, and kept within narrow ranges of pH. Within you, the water in your blood, plasma, and cells is a quiet echo of those primordial oceans. On a planetary scale, water connects all living things. It cycles endlessly through evaporation, condensation, and precipitation. The water you drink tonight may have once flowed through rivers, fallen as snow, or drifted in clouds. It may have been part of glaciers, seas, or the cells of ancient plants and animals. The molecules themselves are timeless, endlessly recycled, passed from one form of life to another. In this way, water is a thread that ties together past, present, and future. And so, when you think of water, you may see more than a familiar liquid. You may see the universal solvent, the gentle stabilizer, the carrier of nutrients, the regulator of heat, the medium of reactions, and the cycle of continuity. It is both simple and profound, both ordinary and miraculous. It sustains every breath, every heartbeat, every moment of life. As you rest, imagine yourself floating in water, calm, supported, weightless. Just as water carries the molecules of life, it also carries you. Its bonds are quiet but strong. Its flow is gentle but unceasing. Water whispers the chemistry of life in every droplet, in every wave, in every cell. And as you drift toward sleep, let this thought remain. Within you and around you, water is always present, always moving, always sustaining. 4. Carbon, the framework of life. As we continue our quiet journey through the chemistry of life, let us now turn our attention to one element above all others, the element that serves as the backbone, the architect, and the unifying thread of biology, carbon. Carbon is not the most abundant element in the universe. Hydrogen and helium claim that honor, making up the majority of stars. Even on Earth, oxygen and silicon are more plentiful overall. And yet, when it comes to life, carbon holds a position of extraordinary importance. Every protein, every carbohydrate, every lipid, and every nucleic acid, in short, every molecule that defines living systems is built on a foundation of carbon atoms. To study biochemistry is to study the endless creativity of carbon, arranging itself into forms both simple and unimaginably complex. Why carbon? The answer lies in its quiet, versatile nature. Carbon sits at the center of the periodic table's second row, with 4 electrons in its outer shell. This means it has the ability to form 4 covalent bonds with other atoms. Unlike many elements, which are limited in their bonding patterns, carbon is open and flexible. It can link with 1, 2, 3, or 4 partners. It can form single bonds, double bonds, or even triple bonds. It can connect to other carbons, creating chains, rings, and branching structures. In essence, carbon is a builder, capable of weaving endless patterns out of a limited set of tools. Imagine carbon as a set of hands, 4 in number, reaching outward in search of connection. With these hands, carbon can hold hydrogen atoms, creating simple hydrocarbons like methane. With other carbons, it can form long chains, like those that compose fatty acids. By linking in closed loops, it creates rings, like those found in glucose and other sugars. And with double bonds, it introduces flexibility and variation, altering the shapes of molecules in subtle ways. From this quiet ability to bond in multiple directions arises the vast diversity of organic chemistry. One of the simplest examples of carbon's creativity is the family of molecules known as hydrocarbon structures composed only of carbon and hydrogen. Though basic in composition, hydrocarbons demonstrate carbon's range. Methane, with 1 carbon and 4 hydrogens, is a tiny, stable molecule. But extend the chain, and you find ethane, propane, butane, and beyond. Bend the chain into a ring, and you find cyclohexane. Add double bonds, and new possibilities emerge. These molecules form the backbone of fuels, fats, and many cellular structures. Yet hydrocarbons are only the beginning. Life depends on carbon's ability to form bonds not only with hydrogen, but also with oxygen, nitrogen, phosphorus, and sulfur. When carbon binds with oxygen and hydrogen, it creates carbohydrates, molecules that store and release energy. When it binds with nitrogen, it creates the amino acids that link into proteins, the workers of the cell. With phosphorus, it creates the backbone of DNA and RNA, the molecules of genetic information. And with sulfur, it forms disulfide bridges, which stabilize the three-dimensional structures of proteins. Carbon does not work alone, it collaborates, weaving itself into larger frameworks with other essential elements. One of carbon's most elegant properties is its ability to form isomers molecules with the same chemical formula, but different structures. Glucose, fructose, and galactose, for instance, all share the formula CHO. Yet their arrangements differ, giving them distinct properties. This simple principle allows for incredible diversity. By rearranging bonds and orientations, carbon can produce molecules that behave differently, interact differently, and serve different purposes. The subtle difference between two isomers can mean the difference between nourishment and toxicity, between energy and waste. Carbon's versatility also extends to the realm of macromolecules. Consider the molecule of DNA, the great archive of life's instructions. At its core, DNA is a chain of nucleotides, each built upon a sugar made of carbon atoms arranged in a ring. The backbone of the DNA helix is a repeating pattern of carbon-containing sugars linked to phosphate groups. From this structure arise the bases adenine, thymine, guanine, and cytosine, themselves built upon carbon rings. Without carbon's ability to form stable yet flexible bonds, DNA's double helix could not exist. Proteins, too, reveal carbon's central role. Each amino acid, the building block of proteins, is constructed around a central carbon atom. This carbon holds 4 attachments: an amino group, a carboxyl group, a hydrogen, and a variable side chain. It is the side chain, sometimes as simple as a single hydrogen, sometimes as complex as a long aromatic ring, that gives each amino acid its character. But at the center, always, is carbon, serving as the point of connection. Lipids, the molecules that form membranes and store energy, are likewise carbon-based. Long chains of carbon and hydrogen create fatty acids, which assemble into phospholipids. These molecules, with their water-loving heads and water-fearing tails, line up to form the bilayer of cell membranes. Here again, carbon's ability to form long, stable chains makes structure possible. Without carbon, the barrier that separates life from non-life, the cell membrane, could not exist. Even at the smallest scale, carbon plays a role in the transfer of energy. The molecule adenosine triphosphate, or ATP, is often called the energy currency of the cell. Its structure is built upon a carbon-containing sugar, ribose, linked to nitrogenous adenine and 3 phosphate groups. When ATP releases energy, it is the carbon framework of ribose and adenine that holds the molecule together, making its energy transfers possible. Beyond its structural importance, carbon also connects life to the larger environment through the carbon cycle. Plants capture carbon dioxide from the air, using sunlight to convert it into sugars. Animals consume those sugars, breaking them down to release energy, and in the process, exhaling carbon dioxide back into the atmosphere. Microorganisms in the soil and oceans recycle carbon in countless ways, returning it to circulation. Through this cycle, Carbon atoms move continuously between air, water, soil, and living organisms. The carbon in your body tonight may once have drifted as gas in the atmosphere, rested in the leaf of a tree, or flowed through the blood of another animal. This cycle has been operating for billions of years, steady and unbroken. Carbon does not disappear, it simply changes form. It is taken in, transformed, and released over and over. In this way, carbon reminds us of continuity, the quiet truth that all living beings share the same materials, passed down and reused across generations and eras. When you reflect on carbon, it is helpful to remember that its presence is not limited to grand structures like DNA or proteins. It is in every breath you take, in the sugars that sweeten fruit, in the oils that nourish seeds, in the wood of trees, in the fibers of your muscles. Carbon surrounds you, supports you, and composes you. Without it, life would be impossible. With it, life has flourished in uncountable forms, from the simplest microbes to the most complex ecosystems. And so, as we rest with carbon, we see not only an element, but a storyteller. Each bond it forms, each chain it builds, each ring it shapes, is part of the great narrative of life. Quiet, versatile, enduring carbon is the framework upon which biology is written. 5. The 4 families of biomolecules. As we move further into the chemistry of life, we arrive at the point where atoms and simple molecules come together into larger, more complex structures. These are the great families of biomolecules, the essential substances from which all living beings are built. Though they are many in number and varied in form, they can be grouped into 4 broad categories: carbohydrates, lipids, proteins, and nucleic acids. Each of these families contributes something different to life, yet together they form the complete foundation of biology. They are the materials from which cells construct themselves, the fuels that provide energy, the tools that perform work, and the archives that preserve information. The first family, the carbohydrates, are molecules built of carbon, hydrogen, and oxygen, often in the ratio that resembles water bound to carbon. At their simplest, they are small sugars such as glucose and fructose, each one a ring of carbon atoms carrying groups of oxygen and hydrogen. These tiny molecules are sweet to the taste and immediate in their effect. When dissolved in the bloodstream, glucose becomes the principal source of quick energy for cells. With every breath you take, oxygen enters your body, and glucose, broken down step by step, is combined with that oxygen to release energy in a controlled fashion. The result of this process is adenosine triphosphate, the small packets of chemical energy that fuel muscles, nerves, and every reaction within you. Sugars can also link together, 2 at a time, to form molecules like sucrose, or in long chains to form starch and glycogen. In these larger forms, carbohydrates become stores of energy, reserves to be drawn upon when the immediate supply runs low. In plants, another carbohydrate, cellulose, gives strength to cell walls, forming the sturdy yet flexible framework that allows trees to grow tall and grasses to bend in the wind. Even though we cannot digest cellulose, it passes through our systems as fiber, supporting the quiet work of digestion. The second family, the lipids, are more subtle. They do not dissolve easily in water, and this gives them a character all their own. They are nature's reservoirs and protectors. Many lipids take the form of fats, which are built from a glycerol backbone joined to 3 long chains called fatty acids. These chains are mostly carbon and hydrogen, holding dense stores of chemical energy. Gram for gram, fats carry more than twice the energy of sugars, making them excellent reserves for long-term storage. In animals, they also provide insulation, keeping bodies warm, and cushioning, protecting organs from shock. In plants, oils serve as food stored in seeds, awaiting the moment when a new plant will grow. Another type of lipid, the phospholipid, plays a quieter but equally important role. Each phospholipid has two parts, a head that is drawn to water and a tail that avoids it. When many come together, they naturally arrange into layers, with the heads facing water and the tails tucked inward. In this way, they form membranes, thin yet resilient sheets that enclose cells and separate the inner from the outer. Without these membranes, life could not create order, and the delicate chemistry within cells could not be maintained. Lipids also form rings, as in steroids. Cholesterol, a familiar example, helps stabilize membranes and serves as the starting point for hormones that guide growth, metabolism, and reproduction. In this way, lipids are not only stores of energy but also architects of structure and messengers of information. The third family, proteins, are perhaps the most versatile. Built from smaller units called amino acids, they are long chains that fold into intricate shapes. Each amino acid has a central carbon atom holding four groups: an amino group, a carboxyl group, a hydrogen atom, and a variable side chain. The side chain gives each amino acid its character, and when many are linked, the chain bends and folds, guided by attractions and repulsions among its parts. From this folding comes shape, and from shape comes function. Proteins serve as enzymes, catalysts that allow life's reactions to occur at the right speed and under the right conditions. Without enzymes, the breakdown of food, the copying of DNA, and the countless other reactions of life would be too slow to sustain existence. Proteins also provide structure, forming the fibers of muscles, the keratin of hair and nails, and the collagen of skin and connective tissue. They carry signals, as in the receptors that span cell membranes, and they defend the body, as in the antibodies that recognize and neutralize invaders. In short, proteins are the workers of life, each one specialized, each one shaped for a task, each one indispensable. The fourth family, nucleic acids, hold life's instructions. These molecules, DNA and RNA, are chains of nucleotides, and each nucleotide is composed of three parts: a sugar, a phosphate group, and a nitrogenous base. The sugar is a small carbon ring. The phosphate provides a link, and the base, one of four possible kinds, carries the information. In DNA, the bases are adenine, thymine, cytosine, and guanine. They pair with each other, adenine with thymine, cytosine with guanine, forming the rungs of a double helix. This elegant spiral, discovered in the middle of the 20th century, is the archive of life's memory. Every trait, every protein, every process of growth and repair is coded in the sequence of bases. RNA, a close relative of DNA, carries these instructions outward, guiding the construction of proteins in the ribosomes of cells. Without nucleic acids, there could be no inheritance, no continuity from one generation to the next, no unfolding of life's story across time. When we step back, we see that these four families, carbohydrates, lipids, proteins, and nucleic acids are not isolated. They interact and overlap. Carbohydrates and lipids provide energy. Proteins carry out the work, and nucleic acids hold the instructions. But carbohydrates are also attached to proteins on cell surfaces, serving as markers of identity. Lipids form membranes in which proteins are embedded, shaping communication and transport. Nucleic acids direct the making of proteins, which in turn regulate carbohydrates and lipids. Nothing stands alone. Life is built on cooperation among these molecules, each family supporting and depending on the others. Perhaps the most soothing thought is that all of this intricacy comes from such simple beginnings. Carbon atoms linking to hydrogen, oxygen, nitrogen, phosphorus, and sulfur, guided by the quiet laws of chemistry, give rise to molecules of sweetness, strength, structure, and memory. Out of these four families flows every heartbeat, every breath, every moment of thought and rest. They are the gentle architecture of life, endlessly repeating, endlessly renewing, yet always forming something new. And so, as you drift deeper into relaxation, you may reflect on these families not as abstract categories, but as the very fabric of your being. The glucose that fuels your mind, the lipids that form your cells, the proteins that carry your breath, the DNA that records your history. They are within you now, working quietly, tirelessly, invisibly. They are the four families of life, the foundation upon which everything rests, the chemistry that never sleeps, sustaining you through the night and into tomorrow. 6. Carbohydrates. In detail, we have already seen that life rests on four great families of biomolecules, and among them, carbohydrates are perhaps the most immediate in their effect. They are the fuels of movement and thought, the first molecules the body reaches for when it needs energy quickly. Yet they are also more than this. They are not only sources of nourishment, but also materials for building, for storing, and for connecting one cell to another. To linger with carbohydrates is to understand the simple sweetness of life's chemistry and the quiet strength it provides. Carbohydrates are built from carbon and oxygen. Their name itself reflects this, for they were once described as hydrates of carbon. Their simplest units are called monosaccharides, meaning single sugars. These are small molecules, most often containing 5 or 6 carbon atoms arranged in a ring. Glucose is one of the most familiar, and it is central to nearly all forms of life. In plants, glucose is created by the capture of sunlight through photosynthesis, a quiet process that transforms carbon dioxide and water into sugars and oxygen. In animals, glucose enters through food, carried in the bloodstream to every corner of the body. When a muscle contracts, when a neuron fires, when a cell divides, it is glucose that provides the energy. Another important monosaccharide is fructose, which is naturally present in fruits and honey. Its arrangement differs slightly from glucose, but the body can convert it easily for energy. Galactose, too, is a simple sugar, often found as part of larger molecules. These monosaccharides are sweet to the taste and small enough to move quickly across membranes and into cells. When two monosaccharides join, they form disaccharides. One familiar example is sucrose, the common table sugar that combines glucose and fructose. Another is lactose, found in milk, which links glucose and galactose. A third, maltose, is produced when starch is broken down. These disaccharides can be split apart during digestion, releasing the individual monosaccharides that can then be used for energy. Carbohydrates reveal their full importance when many monosaccharides join together into long chains, forming polysaccharides. Here, the same simple units create great variety. Starch, for instance, is the way plants store glucose. In grains, roots, and tubers, starch accumulates as a reserve of energy. When consumed, enzymes break it down step by step, releasing glucose molecules that can fuel metabolism. Animals, including humans, store glucose in a similar way, but the storage form is called glycogen. It is found in the liver and in muscles, ready to be mobilized when the body needs a sudden supply of energy. Glycogen is especially important in activities that require bursts of effort, as it can be broken down quickly into glucose. Another polysaccharide, cellulose, is not used for energy but for structure. It is composed of glucose units, just like starch and glycogen, but they are linked in a different way. This difference gives cellulose a rigidity and strength that no enzyme in the human body can easily break. In plants, cellulose forms the walls of cells, providing support for stems, leaves, and wood. Without cellulose, plants could not stand upright, forests could not grow, and the air itself would be different, for tall trees help regulate the balance of gases in the atmosphere. Though we cannot digest cellulose, it passes through our bodies as fiber, and in doing so, it aids digestion, helping food move smoothly through the system. Other animals, such as cows and termites, host microbes that can digest cellulose, allowing them to draw energy from grasses and wood. In this way, cellulose plays a role not only in structure, but also in ecological cycles of energy. Beyond these common forms, carbohydrates also serve more specialized purposes. Some are attached to proteins and lipids on the surfaces of cells, creating patterns that act like identification cards. These carbohydrate markers help the immune system recognize the difference between the body's own cells and foreign invaders. They also allow cells to communicate, to recognize one another, and to form tissues. Others act as components of nucleotides, the building blocks of DNA and RNA. In these cases, the sugar is not primarily a source of energy, but a part of the framework that carries genetic information. The way carbohydrates function in metabolism is both simple and elegant. When glucose enters a cell, it undergoes a series of steps known as glycolysis. This pathway breaks the 6-carbon sugar into 2 smaller molecules, releasing a modest amount of energy. The products are then carried into the mitochondria, the cell's powerhouses, where they enter the citric acid cycle and the electron transport chain. With each stage, more energy is released and captured in the bonds of ATP. At the end, the carbon atoms of glucose are released back into the world as carbon dioxide, ready to be taken up by plants and recycled once again into sugars. Thus, every breath you exhale is a quiet reminder of this ongoing exchange, this gentle circle of matter and energy. The role of carbohydrates extends even further into the rhythms of daily life. After a meal, blood glucose rises, and the hormone insulin helps carry it into cells, storing some as glycogen for later. Between meals, or during sleep, glucose levels fall, and the body draws on those reserves, keeping the mind and muscles supplied with fuel. This balance is maintained with remarkable precision, an invisible harmony that continues without effort or awareness. Carbohydrates also provide comfort in more subtle ways. The taste of sweetness is one of the first sensations recognized by infants, a signal of nourishment and safety. Across cultures, Sweet foods are shared in celebrations, rituals, and moments of rest. Though biochemistry explains their energy, the human experience of carbohydrates carries warmth and connection. They are reminders of abundance and continuity, woven into the fabric of life, both scientifically and culturally. When you reflect on carbohydrates, you can see them as more than molecules. They are the morning energy that allows a bird to take flight. They are the reserves that let a tree bloom in spring after a long winter. They are the steady supply that fuels your own thoughts, movements, and dreams. From the smallest monosaccharide to the largest polysaccharide, carbohydrates carry with them the quiet strength of sustenance. And so, as you rest, you may picture carbohydrates as threads of sweetness weaving through every living system. They are drawn from the sunlit air by plants, shared through food, carried in blood, and finally returned as breath. They move gently, endlessly, sustaining the rhythm of life. In their simplicity lies a profound truth, that life itself is nourished by small, repeating units, patiently linked together, endlessly recycled, always present. They are the chemistry of energy and the chemistry of continuity. 7. Lipids in detail. If carbohydrates can be seen as the quick sparks of energy, lipids may be thought of as the slow-burning lamps of life. They are molecules that store energy quietly, provide structure to cells, and participate in signaling between tissues. Unlike sugars, they are not immediately soluble in water. This single characteristic shapes all of their roles. For in the watery environment of the body, a substance that resists dissolving takes on a different kind of purpose. It forms boundaries, reservoirs, and messages, all of which life requires to remain balanced and whole. Lipids come in many forms, but their common feature is the presence of long chains or rings of carbon and hydrogen. This makes them hydrophobic or water-fearing, though many also have parts that interact with water. Their diversity is great, yet their contributions to life can be gathered into three main areas. They serve as stores of energy, they provide the architecture of membranes, and they act as signaling molecules. Perhaps the most familiar lipids are fats and oils. These molecules, known more formally as triglycerides, are composed of a glycerol backbone bound to 3 fatty acids. The fatty acids themselves are long chains of carbon and hydrogen, sometimes containing double bonds. If all the bonds between carbons are single, the chain is said to be saturated, and the molecules pack closely, forming solids at room temperature. Butter and animal fats are familiar examples. If double bonds are present, the chain bends slightly, preventing tight packing. These are unsaturated fats, and they are liquid oils, flowing easily as in olive oil or sunflower oil. In either case, the long chains of carbon store a remarkable amount of energy. Gram for gram, lipids hold more than twice the energy of carbohydrates. This makes them efficient reserves, well suited for long-term storage in the body. Beyond storing energy, lipids also protect and insulate. Fat beneath the skin cushions delicate organs, provides warmth in cold environments, and serves as a quiet buffer against sudden shocks. In animals that hibernate or those that endure long migrations, fat is a trusted reserve, drawn upon slowly when food is scarce. In seeds and nuts, oils store energy for the growing plant that will one day emerge. These reserves are patient, waiting until the moment of need, and then yielding their energy gently and steadily. Yet lipids are not only about storage, they are also about structure. Among the most elegant molecules in biology are the phospholipids. These are similar to triglycerides, but instead of 3 fatty acid chains, they contain 2, along with a phosphate group attached to glycerol. The phosphate carries a negative charge, making it hydrophilic or water-loving. The fatty acid tails remain hydrophobic. This dual nature creates a molecule with 2 personalities, a head that seeks water and tails that avoid it. When many phospholipids gather in water, they naturally arrange themselves into bilayers. The heads face outward, touching water, while the tails hide inward, away from it. This simple arrangement gives rise to membranes, thin sheets that surround cells and enclose their contents. The membrane is not a rigid wall, but a fluid mosaic, allowing movement and flexibility. Proteins float within it, sugars attach to its surface, and ions pass through channels. It is the barrier that separates inside from outside, yet it is selectively open, allowing nutrients in and waste out. Every cell, from the simplest bacterium to the most complex human neuron, relies on membranes built from phospholipids. Without them, life's delicate chemistry could not be contained, and order would dissolve into chaos. In this way, lipids provide not only storage but also boundaries, shaping the very identity of living cells. Lipids also take the form of rings, creating molecules called steroids. One of the most familiar is cholesterol, which often carries an association with health in modern thought. Yet, in its essence, cholesterol is not a threat but a necessity. It is part of membranes, lending them stability and fluidity. Without it, membranes would be too rigid or too fragile, unable to function properly. Cholesterol is also the starting point for steroid hormones, a class of signaling molecules that guide growth, metabolism, and reproduction. Hormones such as cortisol, estrogen, and testosterone all arise from the same carbon framework of cholesterol, subtly modified to carry different messages. These hormones act at a distance, moving through the bloodstream to influence tissues far from where they were made. In this way, lipids serve as chemical messengers, carrying information quietly from one part of the body to another. Even within the nervous system, lipids play silent roles. Myelin, the insulating sheath that wraps around nerve fibers, is rich in lipids. It allows electrical signals to travel quickly and efficiently along neurons, supporting thought, memory, and movement. Without this lipid-based insulation, communication within the brain and body would be slow and unreliable. Here again, lipids provide not just storage or structure, but function at the deepest level. The metabolism of lipids is slower than that of carbohydrates, but it is equally important. When food is plentiful, excess carbohydrates can be converted into fat, stored for times when food may be scarce. When reserves are needed, lipids are broken down through processes that release their stored energy, supplying ATP to muscles and organs. This quiet cycle has sustained animals through feast and famine, migration and hibernation, growth and rest. Lipids also participate in ecological cycles. The waxy cuticle that coats the leaves of plants, preventing water loss, is made of lipids. The oils and seeds nourish animals that spread them to new places. In marine environments, tiny organisms produce lipid-rich oils that provide food for countless creatures, forming the base of oceanic food webs. In every corner of life, from the smallest microbe to the largest mammal, lipids are present, supporting, protecting, and sustaining. It is striking to reflect that lipids, so different from sugars in their properties, are built from the same elements, carbon, hydrogen, and oxygen. The difference lies not in what they contain, but in how those atoms are arranged. By forming long nonpolar chains instead of balanced rings, lipids take on their unique resistance to water. From this simple chemical truth arises a vast world of functions, energy reserves, membranes, hormones, insulation, and more. As you rest, you may imagine lipids as the quiet layers beneath the surface of life. They are the soft cushions of energy, the unseen walls that hold cells together, the signals that pass through blood and tissue, the insulation that allows thought to move swiftly. They do not announce themselves in taste as sugars do. They do not fold into intricate shapes as proteins do. Instead, they work silently, forming the background architecture, the stable reserves, the patient keepers of energy and form. In their silence lies their strength. Lipids remind us that much of life's chemistry does not call attention to itself. It works quietly, reliably, waiting until needed, always present but seldom noticed. And so, as you sink further into stillness, you may reflect on the lipids within you, in your membranes, in your reserves, in your very thoughts, carrying out their steady, essential work without fanfare, yet making life possible in every moment. 8. Proteins, in detail. Among the 4 families of biomolecules, proteins are perhaps the most versatile and dynamic. If carbohydrates provide quick bursts of energy, and lipids serve as quiet reserves and boundaries, proteins can be thought of as the workers, the builders, and the caretakers of life. They are not only the most abundant biomolecules within cells, but also the most varied in their shapes and functions. Wherever there is life, there is protein folding, moving, binding, and transforming. Every heartbeat, every thought, every breath depends on their subtle activity. Proteins are made of smaller building blocks called amino acids. There are 20 common amino acids used in nearly all forms of life. Each amino acid shares the same basic structure: a central carbon atom bonded to 4 groups. One is a hydrogen atom, another is an amino group carrying nitrogen. A third is a carboxyl group, which acts as an acid. The fourth, however, is what makes each amino acid unique. It is a side chain, also called the R group, which may be as simple as a single hydrogen or as complex as a long aromatic ring. The properties of these side chains, whether they are charged or neutral, hydrophilic or hydrophobic, determine how the amino acids interact with one another. When amino acids are joined in long chains through peptide bonds, they form polypeptides, and when these polypeptides fold into 3-dimensional structures, they become proteins. The sequence of amino acids in a chain is called the primary structure. Like letters in a sentence, this sequence determines everything that follows. From this foundation, local patterns arise: coils known as alpha helices and sheets known as beta-pleated sheets. These patterns are held together by hydrogen bonds and form the secondary structure. Further folding and twisting, driven by the interactions among side chains, create the tertiary structure, the overall shape of a single protein molecule. In some cases, multiple polypeptides join together, forming a quaternary structure. Hemoglobin, the protein that carries oxygen in the blood, is a classic example, consisting of 4 subunits working together in harmony. The shape of a protein is crucial, for structure dictates function. An enzyme, for instance, has a pocket called an active site. This pocket is shaped to fit only a specific molecule, called the substrate. When the substrate binds, the enzyme catalyzes a reaction, lowering the energy required and allowing the process to occur at a pace suitable for life. Without enzymes, the reactions that sustain metabolism would be so slow that life could not exist. With them, reactions proceed millions of times faster, all under gentle conditions of temperature and pH. Proteins also serve as transporters. Hemoglobin, as mentioned, carries oxygen from the lungs to tissues. Other proteins shuttle ions across membranes, maintaining the balance of sodium and potassium that allows nerve impulses to fire. Some proteins act as storage molecules, such as ferritin, which stores iron within cells until it is needed. Others act as receptors embedded in cell membranes, detecting signals from hormones or neurotransmitters and relaying them inward to trigger a response. The structural roles of proteins are equally important. In muscles, actin and myosin proteins interact to allow contraction and movement. In skin, hair, and nails, keratin provides strength and resilience. In connective tissues, collagen forms a network that supports and binds cells together, giving form and elasticity to tendons, ligaments, and skin. These structural proteins are not as dynamic as enzymes, but they are no less vital, for they give bodies their shape and endurance. Proteins also play a defensive role. Antibodies, produced by the immune system, are proteins designed to recognize and bind foreign invaders. Each antibody has a unique shape that allows it to attach to a particular antigen, marking it for destruction or neutralization. This quiet defense system works continuously, recognizing viruses, bacteria, and other threats, protecting the body from harm. The diversity of proteins is astonishing. Within a single human cell, there may be tens of thousands of different proteins, each with its own role. The variety arises from the nearly endless combinations of the 20 amino acids arranged in chains of varying length. Even a small protein of 100 amino acids has more possible sequences than there are atoms in the universe. Life does not use them all, of course, but the possibilities allow for extraordinary specialization. Proteins are not permanent fixtures. They are constantly being made and broken down. Some last only minutes before being replaced, while others endure for months or years. The balance between synthesis and degradation is finely tuned, ensuring that cells adapt to changing conditions. In times of growth, more proteins are built. In times of stress, damaged proteins are dismantled and recycled. The cell is never static. It is a dynamic workshop where proteins are continually renewed. The process of making proteins, known as protein synthesis, is itself a marvel. The instructions are written in DNA, stored safely in the nucleus. These instructions are transcribed into RNA, which carries the message outward to the ribosomes. There, the RNA is read like a code, each 3-letter sequence specifying one amino acid. The ribosome assembles the amino acids in order, forming the growing chain, which folds into shape as it emerges. In this way, the information stored in nucleic acids is translated into the working molecules of life. Proteins are also sensitive to their environment. Because their shapes are held together by weak interactions, they can be disrupted by changes in temperature, pH, or chemical exposure. When this happens, a protein may unfold, a process known as denaturation. Denatured proteins lose their function, for their shape is essential to their role. This is why high fevers can be dangerous, or why cooking an egg turns its clear proteins into solid white. Within the body, cells work constantly to protect and maintain their proteins, using helper molecules called chaperones to ensure proper folding. It is comforting to think of proteins not only as molecules but as quiet workers, each one carrying out its task faithfully. They do not rest, they do not choose, yet they sustain every function of life. They move oxygen to your cells, they contract your muscles, they digest your food, they mend your tissues, they remember your past encounters with illness. In every moment, they are at work, silent and unseen. And when proteins complete their tasks, they are recycled. Their amino acids are taken apart and reused to build new proteins, forming an unbroken cycle. The nitrogen within them passes through food webs, from soil to plant, from plant to animal, from one generation to the next. In this way, proteins remind us that nothing in life is truly wasted. Everything is transformed. So as you drift further into stillness, you may think of proteins as caretakers within you, folding quietly, working gently, shaping and sustaining your being. They are the silent orchestra of life, playing countless melodies at once, each note essential, each harmony seamless. Without them, life could not exist. With them, life flourishes in infinite forms, delicate, resilient, and enduring. 9. Nucleic acids. In detail, of all the families of biomolecules, nucleic acids are perhaps the most profound. Carbohydrates provide energy, lipids store and protect. Proteins work and build, but nucleic acids preserve the memory of life itself. They are the archives, the instructions, the blueprint written not in ink but in sequences of bases, carried faithfully from one generation to the next. Every cell in your body, every leaf on every tree, every bacterium in the soil contains these molecules. Without them, life would have no continuity, no inheritance, no ability to grow in form and complexity. With them, life is not only sustained, but remembered. Nucleic acids come in two main forms: deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. Both are polymers, long chains built from smaller units called nucleotides. Each nucleotide has three parts. The first is a sugar, a small carbon ring that provides the backbone. In DNA, this sugar is deoxyribose. In RNA, it is ribose, which has one extra oxygen atom. The second part is a phosphate group, which links the sugars together, creating a sturdy chain. The third part is a nitrogenous base, and it is here that the language of nucleic acids is written. In DNA, there are 4 bases: adenine, thymine, cytosine, and guanine. In RNA, thymine is replaced by uracil. These bases are the letters of life's alphabet, simple in number, yet vast in possibility. What makes DNA remarkable is not only the presence of these bases, but the way they pair and order themselves. Adenine pairs with thymine, and cytosine pairs with guanine, each held together by hydrogen bonds. 2 chains of nucleotides, aligned and paired, twist around one another to form the famous double helix. This shape, elegant and simple, is also resilient. It can be opened, copied, repaired, and condensed, yet it always returns to the spiral form, a graceful coil that carries the code of life. The sequence of bases along DNA is the message. Like letters forming words, the order of bases forms instructions. These instructions are divided into units called genes. A gene is not a physical object, but rather a length of DNA whose sequence contains the recipe for a protein. Within the nucleus of a cell, these recipes rest, curled within chromosomes. The human genome, the complete set of instructions for a human being, contains billions of bases, arranged in a sequence that spans volumes beyond imagination. And yet, from this sequence, your body is guided in every detail, from the structure of your bones to the rhythm of your heartbeat, from the color of your eyes to the proteins that heal your wounds. RNA plays a different but equally important role. If DNA is the archive, RNA is the messenger. When a cell needs a particular protein, the relevant gene is transcribed into a strand of RNA. This messenger RNA leaves the nucleus and travels to a ribosome, where its sequence is read. Each group of 3 bases corresponds to 1 amino acid, and the ribosome uses this code to assemble a protein. In this way, information written in DNA is translated into the workers and builders that keep life alive. RNA also takes other forms. Transfer RNA carries amino acids to the ribosome. Ribosomal RNA forms part of the ribosome itself, providing structure and catalysis. Small RNAs regulate which genes are expressed, turning them on or off in response to signals. Together, these forms of RNA make the flow of information possible. The stability of nucleic acids allows for continuity, but their ability to change allows for diversity. Occasionally, errors occur when DNA is copied. A base may be substituted, added, or lost. Most changes are silent, having little effect. Some are harmful, disrupting the function of a protein, yet some provide variation, offering the raw material for evolution. Across generations, these variations accumulate, and from them, the diversity of life arises. Thus, nucleic acids are both faithful and flexible. They preserve the past while allowing for the possibility of the future. Nucleic acids also carry a quiet poetry in their cycle of inheritance. Half of your DNA came from one parent, and half from the other. You carry within you sequences that stretch back through countless generations, shared with ancestors you will never meet, connected to relatives you will never know. In every cell, your DNA is both personal and universal, a story that began long before you and will continue after you. It ties together all living beings, for the genetic code is nearly the same across species. The bases that spell out a protein in a bacterium are the same as those that spell out a protein in a human. Life speaks a shared language, written in the letters of nucleic acids. On a molecular level, nucleic acids are active participants in the rhythm of the cell. They are copied during cell division, ensuring that new cells receive the same instructions. They are repaired when damaged, using one strand as a template to restore the other. They are wound and unwound, condensed into chromosomes during division and relaxed during expression. Though they seem like silent archives, they are constantly in motion, responding, adjusting, and maintaining integrity. It is humbling to reflect that the instructions for life, so intricate and vast, are carried in a molecule built from such simple parts: a sugar, a phosphate, and a base. 4 bases, repeated billions of times, are sufficient to create the vast complexity of living forms. From these sequences arise the wings of birds, the roots of trees, the minds of humans, the songs of whales, the quiet persistence of microbes. All are variations of the same language, spoken across the biosphere. As you drift into deeper rest, you may imagine nucleic acids as threads of a great tapestry, woven into spirals of information. They coil and uncoil. They copy and repair. They instruct and guide. They are within you, within every cell, holding the memory of your past and the potential of your future. They are at once fragile and enduring, vulnerable to change, yet strong enough to persist across billions of years. In their quiet spirals, life finds its continuity, its variety, and its resilience. And so, nucleic acids remind us of something gentle and eternal. Life is not a moment, but a flow, not a single note, but a melody passed across time. DNA and RNA are the instruments on which this melody is played, carrying forward the song of existence. Tonight, as you rest, they continue their work within you, unseen but unceasing, whispering the instructions of life into every corner of your being. 10. Enzymes and catalysis. Life depends on chemistry, yet the natural pace of chemical reactions in ordinary conditions is often too slow to sustain it. Many of the transformations that keep cells alive, the breakdown of food, the replication of DNA, the movement of ions would take hours, days, or even years to occur on their own. But life cannot wait. It requires a rhythm, a flow, a pace measured in seconds and fractions of seconds. To make this possible, organisms rely on special proteins called enzymes. These are the gentle catalysts of life, speeding reactions without being consumed, guiding molecules toward transformation while remaining unchanged themselves. An enzyme is a protein, and like all proteins, its power comes from its shape. Within its folded form lies a small pocket called the active site. This pocket is not random. It is shaped precisely to fit the molecules that will bind to it. When a substrate enters an active site, it is held in place by weak forces, hydrogen bonds, ionic attractions, van der Waals interactions. The fit may be snug, like a key in a lock, or it may involve slight adjustments as the enzyme molds around its partner. This model of gentle adjustment is often called the induced fit, and it highlights the flexibility of enzymes. Once the substrate is bound, the enzyme lowers the activation energy of the reaction. Activation energy is the small hill that reactants must climb before they can be transformed into products. By stabilizing the transition state, the enzyme makes the hill smaller, easier to cross. The reaction proceeds more quickly, yet the enzyme itself is not consumed. When the product is released, the enzyme is free to bind another substrate, ready to catalyze the reaction again and again. Some enzymes can process thousands of molecules per second, tirelessly repeating the same task with precision. The diversity of enzymes is astonishing. There are enzymes that cut bonds, enzymes that join molecules together, enzymes that rearrange atoms, and enzymes that transfer groups from one molecule to another. Digestive enzymes, for instance, break down the food we eat. Amylase, present in saliva, begins the digestion of starch into sugars as soon as food enters the mouth. Proteases break proteins into amino acids, and lipases split fats into glycerol and fatty acids. Without these enzymes, nutrients would pass through the body largely unchanged, unavailable for energy or growth. Other enzymes work deep within cells. DNA polymerase, for example, copies DNA during cell division, ensuring that each new cell receives a complete set of instructions. RNA polymerase transcribes DNA into RNA, the first step in protein synthesis. ATP synthase, located in the mitochondria, manufactures ATP, the energy currency of the cell. by harnessing the flow of protons across a membrane. Each enzyme is specialized, shaped by evolution to recognize particular substrates and carry out particular reactions. Despite their diversity, enzymes share a common quality: specificity. Each one is tuned to act on certain molecules and not others. This specificity arises from the structure of the active site. Even a small change in the sequence of amino acids can alter the shape and function of an enzyme. Sometimes such changes are beneficial, giving rise to new abilities. Sometimes they are harmful, leading to metabolic disorders. Yet the principle remains. Life's chemistry is controlled with remarkable precision, thanks to the delicate shapes of enzymes. Enzymes also respond to the environment. Their activity depends on temperature, pH, and the presence of cofactors. Most human enzymes, for example, function best around body temperature and near neutral pH. Extremes of heat or acidity can denature the protein, unfolding it and destroying its active site. Yet life adapts. Organisms that live in hot springs or deep-sea vents have enzymes stable at high temperatures. Those that live in acidic environments have enzymes suited to low pH. In every environment where life exists, enzymes have evolved to match the conditions, allowing chemistry to proceed with balance. Some enzymes require partners to function. These partners may be metal ions such as magnesium or zinc, or organic molecules called coenzymes. Vitamins obtained through diet often serve as coenzymes or precursors to them. Without these helpers, many enzymes would remain idle. Together, enzyme and cofactor form a complete catalytic unit, capable of guiding reactions that sustain life. The regulation of enzymes is another quiet layer of control. Cells cannot allow every enzyme to act at full speed all the time. Instead, they use signals to turn enzymes on or off, to increase or decrease their activity. Feedback inhibition is one common mechanism. When the product of a pathway accumulates, it binds to an enzyme earlier in the pathway, reducing its activity. In this way, the pathway slows down, preventing waste. When the product is used up, the inhibition lifts, and the pathway resumes. Such feedback ensures that cells maintain balance, producing neither too much nor too little of what they need. There are also inhibitors molecules that interfere with enzymes. Some bind reversibly, temporarily blocking the active site. Others bind irreversibly, disabling the enzyme permanently. In nature, many toxins act as enzyme inhibitors, disrupting metabolism. Yet inhibitors are not always harmful. In medicine, they are used deliberately to control processes, slowing reactions that cause disease. The same principle that can harm can also heal, depending on how it is guided. When reflecting on enzymes, it is helpful to see them not as abstract molecules, but as quiet caretakers of life's chemistry. They are the reason that sugar can be turned into energy in seconds rather than years. They are the reason that DNA can be copied faithfully and quickly during cell division. They are the reason that thoughts travel through the nervous system at a pace that allows perception and action. Every instant of life is filled with their activity, silent and invisible, yet essential. It is also humbling to recognize that enzymes work without intent or awareness. They do not know what they do, yet they do it faithfully. They fold into their shapes, they bind their substrates, they catalyze their reactions. They are like patient artisans, repeating the same motion endlessly, each repetition sustaining life. Within every cell of your body, countless enzymes are working at this very moment, transforming molecules, producing energy, repairing damage, maintaining balance. And so, enzymes remind us of the quiet efficiency of nature. Life does not rush blindly. It guides its chemistry with care. Reactions happen not chaotically, but precisely, in the right place, at the right time, at the right speed. This orchestration is possible because of enzymes, the gentle catalysts that make life's complexity possible. As you drift further into stillness, you may imagine enzymes as tiny guardians within you, lowering barriers, smoothing pathways, easing the passage from one state to another. They work quietly, without pause, ensuring that the chemistry of life flows as it should. With every breath, every beat of your heart, every thought that passes through your mind, enzymes are at work, unseen but essential, turning possibility into reality, potential into motion, chemistry into life. 11. Energy in living systems. To understand life is to understand energy. For living things are defined not merely by their structure, but by their activity. A rock and a cell are both made of atoms and molecules, yet the rock is still while the cell is alive. The difference lies in the flow of energy. Life is not static. It is a constant movement of matter and energy through pathways of transformation. Each heartbeat, each breath, each thought is powered by energy, captured, stored, and released with care. Without this flow, cells would cease their functions in moments, and life would quietly end. With it, life continues as a gentle rhythm, sustained hour by hour, day by day. Energy in living systems is carried in many forms, but the most central is a small molecule called adenosine triphosphate, or ATP. ATP is often described as the energy currency of the cell. Just as coins or bills allow exchange in a marketplace, ATP allows energy to be transferred within a cell. It is a compact, portable unit, easy to spend, easy to regenerate. Its structure is simple: a base called adenine, a sugar called ribose, and 3 phosphate groups linked in a row. The bonds between the phosphates, especially the final one, store potential energy. When this bond is broken, ATP releases energy and becomes ADP, adenosine diphosphate. With the addition of another phosphate, ATP is rebuilt again. This cycle of release and regeneration continues endlessly, turning with every breath you take. The cell uses ATP in countless ways. It powers muscle contraction, allowing fibers of actin and myosin to slide past one another so that you may move, stretch, or simply maintain posture as you rest. It fuels the pumping of ions across membranes, establishing gradients that allow nerves to send signals and kidneys to regulate balance. It provides the energy for enzymes to knit together amino acids into proteins, nucleotides into DNA, or fatty acids into lipids. In every domain of life, from bacteria to humans, ATP serves the same role: a tiny packet of usable energy spent in milliseconds and replenished without pause. How, then, is ATP created? The answer lies in metabolism, the sum of all chemical reactions within living systems. Metabolism is not a single process, but a network of pathways, some breaking molecules down, others building them up. These pathways can be divided into 2 broad categories: catabolism and anabolism. Catabolic reactions break larger molecules into smaller ones, releasing energy in the process. The breakdown of glucose, fatty acids, and proteins all fall into this category. Anabolic reactions, on the other hand, use energy to build larger molecules from smaller ones. The synthesis of proteins, DNA, or complex carbohydrates are examples. Catabolism provides the energy, and anabolism uses it, forming a balanced cycle that sustains life. The most familiar catabolic pathway is cellular respiration, in which glucose is broken down step by step to release energy. It begins with glycolysis, a series of reactions in the cytoplasm that splits glucose into two smaller molecules, producing a small amount of ATP. These molecules are then carried into the mitochondria, where they enter the citric acid cycle, also known as the Krebs cycle. Here, carbon atoms are gradually stripped away as carbon dioxide, and high-energy electrons are released. These electrons are passed along a chain of proteins in the mitochondrial membrane, the electron transport chain, where their energy is used to pump protons across the membrane. The protons then flow back through ATP synthase, a remarkable enzyme that spins like a turbine, capturing the flow and using it to rebuild ATP. In this way, the energy once held in glucose is transformed into the universal currency of the cell. Other fuels can also feed into this system. Fatty acids, released from lipids, are broken into two carbon units that enter the citric acid cycle. Amino acids, when not needed for protein, can also be channeled into respiration. Thus, the body is flexible, able to draw energy from multiple sources, each ultimately converted into ATP. This flexibility has allowed life to endure scarcity and abundance, feast and famine, winter and summer, for countless generations. Plants, too, rely on ATP, but they generate it first through photosynthesis. In the chloroplasts, sunlight excites electrons in pigments such as chlorophyll. These high-energy electrons are passed through a chain similar to that in mitochondria, pumping protons and driving ATP synthesis. The energy stored in ATP is then used to build sugars from carbon dioxide and water. In this way, plants capture the light of the sun and transform it into chemical energy, which is then shared with animals, fungi, and microbes when they consume plant matter. The energy that fuels your thoughts and movements tonight was once sunlight, captured by green leaves, stored in sugars, and carried across ecosystems until it reached you. Energy flow in living systems is always coupled. Reactions that release energy are linked to reactions that require it. The breakdown of glucose is linked to the synthesis of ATP. The hydrolysis of ATP is linked to the folding of proteins, the pumping of ions, or the movement of cilia. This coupling ensures efficiency, preventing energy from being lost to heat or wasted on random motion. It is a quiet choreography, molecules passing energy hand to hand like dancers in a circle. Each step timed, each exchange precise. Even at rest, your body is using energy. Cells maintain gradients, repair damage, and replace worn-out molecules. This resting energy use is called the basal metabolic rate, and it represents the silent work of life continuing beneath your awareness. When you move, when you think deeply, when you shiver in the cold, energy use increases. But even in stillness, the quiet consumption of ATP continues, moment by moment, night and day. Energy in living systems is also conserved through storage. When glucose is abundant, some is stored as glycogen in the liver and muscles. When energy intake exceeds immediate need, it is stored as fat, ready for times of scarcity. These reserves represent another rhythm: intake, storage, use, and renewal. Across seasons, across life cycles, organisms rely on stored energy to carry them through. Seeds hold oils to nourish new growth. Bears carry fat through winter sleep. Humans rely on glycogen and fat during fasting. Energy is never wasted. It is held and released as needed, woven into the patterns of survival. As you rest, it is soothing to realize that this flow of energy within you continues without effort on your part. You need not command it. Your cells know what to do. Enzymes act. Gradients shift. ATP is made and spent, endlessly renewed. The energy that sustains your heartbeat and your breath is the same energy that once traveled from the heart of a star, through the warmth of the sun, into the bonds of molecules, and finally into your cells. You are connected to that great flow, not apart from it, but part of it. And so, energy in living systems is more than chemistry. It is the rhythm of life itself. It is the pulse of continuity, the hum beneath every living being. From the tiniest bacterium drifting in the ocean to the largest whale gliding through its depths, energy flows in the same way, captured, stored, released, and renewed. Tonight, as you drift deeper into stillness, you are carried by that same current, a gentle tide of energy moving quietly within you, sustaining you, connecting you to all of life. 12. Cellular respiration. To understand the chemistry of life, it is helpful to follow the path of energy more closely, to trace how the food you eat becomes the quiet fuel for thought, breath, and movement. This path is known as cellular respiration, a series of steps that unfold within your cells every moment of your life. Though the process is intricate, it can be seen as a gentle journey of a single molecule of glucose, broken down carefully, step by step, until its stored energy is released into the form of ATP. Each step is small, deliberate, and beautifully ordered, allowing energy to be captured gradually rather than wasted in a sudden burst. The journey begins with glycolysis, which takes place in the cytoplasm, the watery interior of the cell outside the organelles. The word itself means splitting of sugar. Here, a single 6-carbon glucose molecule is gradually broken into 2 3-carbon molecules called pyruvate. Along the way, a small amount of energy is released. 2 molecules of ATP are gained directly, and high-energy electrons are transferred to a carrier molecule called NADH. Glycolysis requires no oxygen, and so it is ancient, a pathway that likely existed in the earliest forms of life. Even today, it remains essential, serving as the first step in respiration for both aerobic and anaerobic organisms. For you, sitting in quiet rest, glycolysis is happening in countless cells at once, a steady hum of sugar being split and prepared for the next stage. Once glycolysis is complete, the pyruvate molecules are carried into the mitochondria, the organelles often called the powerhouses of the cell. Here, pyruvate undergoes a small transformation. Each is converted into a 2-carbon molecule called acetyl-CoA, with 1 carbon released as carbon dioxide, the same gas that will later leave your body with each exhalation. Along with this change, another molecule of NADH is formed, carrying more electrons to be used later. Acetyl-CoA is the entry ticket into the next cycle, where the true unfolding of respiration occurs. This next stage is the citric acid cycle, sometimes called the Krebs cycle, named after the scientist who described it. Within the mitochondrial matrix, Acetyl-CoA enters the cycle and is combined with a 4-carbon molecule to form a 6-carbon compound called citrate, the molecule that gives the cycle its name. Through a series of gentle transformations, citrate is reshaped, rearranged, and reduced, releasing 2 molecules of carbon dioxide, which are exhaled from the body. At the same time, more NADH and another carrier, FADH, are created, each holding tightly to high-energy electrons. A small amount of ATP is also produced directly. The cycle then regenerates its starting molecule, ready to accept another acetyl-CoA, ready to continue endlessly as long as fuel is available. The citric acid cycle is not only a source of energy, but also a hub of metabolism. Its intermediates are drawn off for other purposes, providing building blocks for amino acids, nucleotides, and other essential compounds. Thus, it is not only a pathway of breakdown, but also one of renewal, a crossroads where catabolism and anabolism meet. The electrons carried by NADH and FADH now proceed to the final stage, the electron transport chain. This stage takes place across the inner membrane of the mitochondria, a folded surface that provides ample room for the machinery of energy conversion. Here, proteins and cofactors are arranged in a line, passing electrons from one to the next, like runners in a relay. As electrons move down the chain, they release energy, and this energy is used to pump protons across the membrane, creating a gradient. On one side of the membrane, protons accumulate. On the other, they are scarce. This imbalance stores potential energy, much like water held behind a dam. At the end of the chain, the electrons meet oxygen, the final electron acceptor. Oxygen combines with the electrons and protons to form water, a gentle conclusion to the journey. Without oxygen, the chain would halt, electrons would back up, and the entire process would stall. This is why oxygen is so vital to life, why every breath you take is tied directly to the quiet chemistry within your mitochondria. The proton gradient built by the chain now powers the most remarkable enzyme of all, ATP synthase. This enzyme spans the inner membrane and allows protons to flow back across. As they pass through, ATP synthase rotates like a molecular turbine, using the energy of the flow to join ADP and phosphate into new ATP molecules. With every turn, ATP is created in abundance. Most of the ATP your cells use is made in this stage, generated not directly by the breakdown of glucose, but by the careful harnessing of a gradient and the work of this spinning enzyme. From one molecule of glucose, cellular respiration can yield around 30 molecules of ATP. Each one is spent quickly, used for muscle contraction, ion transport, protein synthesis, and countless other tasks, then regenerated again. The cycle continues without pause, sustaining every living moment. When oxygen is scarce, as in intense exercise or in some microbes, cells rely on fermentation instead. After glycolysis, pyruvate is converted into lactate in animals or into ethanol and carbon dioxide in yeast. This allows NADH to release its electrons and recycle back to NAD, enabling glycolysis to continue. The yield is small, only 2 ATP per glucose, but it is enough to sustain life temporarily. Fermentation is a reminder of the adaptability of metabolism, the way life finds a way to keep energy flowing even in difficult conditions. To reflect on cellular respiration is to see the elegance of life's chemistry. Glucose, once sunlight captured by plants, is carefully dismantled, its energy not wasted, but captured, step by step. Electrons travel like quiet messengers, Protons flow like gentle tides. ATP synthase spins like a tiny mill wheel. Oxygen accepts the final burden. Water is formed, and ATP is born anew. It is not chaotic but ordered, not wasteful but efficient, a cycle of transformation that has sustained life for billions of years. As you rest, this process continues within you, in your muscles, in your brain, in every cell. Glucose is being split, electrons are being carried, protons are flowing, ATP is being made. The breath you draw provides oxygen. The food you eat provides fuel, and your cells unite them into energy. You need not direct it. It happens silently, automatically, faithfully. And so, cellular respiration is more than chemistry. It is the heartbeat of life at the molecular level. It is the quiet turning of cycles, the graceful passing of electrons, the steady generation of energy. Tonight, as you drift into stillness, this process carries on within you, unbroken, sustaining your rest and preparing you for tomorrow. 13. Photosynthesis. If cellular respiration is the quiet dismantling of energy, photosynthesis is the opposite. It is the building up, the capturing, the storing of energy at its source. For while animals and fungi and many microbes must consume energy from outside, plants, algae, and certain bacteria perform the remarkable feat of drawing energy directly from sunlight. Through photosynthesis, they convert the radiant energy of the sun into chemical energy stored in sugars. In this way, they form the foundation of nearly all food chains on Earth, supporting not only themselves but also every creature that eats them, and every creature that eats those creatures in turn. Photosynthesis is, in essence, the great opening act of life's chemistry, the beginning of energy's journey through the living world. At the heart of photosynthesis is the chloroplast, the green organelle within plant cells. Inside each chloroplast are stacks of flattened sacs called thylakoids, arranged like coins in columns known as grana. The green color comes from chlorophyll, a pigment that absorbs light most strongly in the blue and red parts of the spectrum, reflecting green. This pigment, along with others such as carotenoids, captures photons of light and transfers their energy into electrons. The process begins with this simple act. A photon from the sun strikes a pigment molecule, and an electron is excited to a higher energy level. This moment, happening countless times in every leaf under sunlight, is the first step in the transformation of radiant energy into chemical energy. The excited electrons travel through a chain of proteins embedded in the thylakoid membrane, a system known as the light reactions. As the electrons move, their energy is used to pump protons across the membrane, creating a gradient. Just as in mitochondria, this gradient is harnessed by ATP synthase, which allows protons to flow back and uses their energy to form ATP. Alongside ATP, another molecule, NADPH, is formed. Both ATP and NADPH are temporary carriers of energy, holding the power of sunlight in chemical form. Oxygen is also released at this stage, a byproduct of splitting water molecules to replace the excited electrons lost by chlorophyll. Each oxygen molecule released into the air carries with it the memory of a photon, a tiny fragment of starlight transformed into breath. This stage, the light reactions, depends on sunlight directly. But photosynthesis also includes a second stage, known as the Calvin cycle, or the light-independent reactions. These take place in the stroma, the fluid surrounding the thylakoids. Here, The ATP and NADPH produced in the light reactions are used to power the fixation of carbon dioxide. Molecules of carbon dioxide, drawn from the air, are attached to a 5-carbon compound called ribulose bisphosphate, or RuBP. This step is catalyzed by an enzyme named Rubisco, one of the most abundant proteins on Earth. The unstable 6-carbon product quickly splits into 2 3-carbon molecules, which are then reshaped, energized, and assembled into glucose and other sugars. These sugars store the energy of sunlight in stable bonds, bonds that can be transported throughout the plant or consumed by animals and microbes. The overall equation of photosynthesis is deceptively simple. Carbon dioxide plus water plus sunlight yields glucose and oxygen. Yet within that simplicity lies extraordinary elegance. Sunlight, ephemeral and fleeting, is captured and stored. Carbon dioxide, invisible and diffuse, is fixed into solid form. Water, abundant and flowing, donates electrons and hydrogen atoms. The result is sugar, a molecule that holds the energy of light in a form that can be shared, and oxygen, a gas that sustains respiration across the biosphere. Photosynthesis does not only sustain plants, it sustains all of us. Every bite of food, every breath of oxygen, is a gift from this process. Even the energy in fossil fuels, ancient reserves of carbon-rich compounds, originated as sunlight captured by photosynthetic organisms millions of years ago. In this way, photosynthesis connects past and present, atmosphere and biosphere, plants and animals, stars and cells. There are variations of photosynthesis that reveal the adaptability of life. Some plants in hot, dry climates use C photosynthesis, which minimizes water loss while capturing carbon dioxide more efficiently. Others use CAM photosynthesis, opening their stomata at night to avoid daytime heat, storing carbon dioxide for use during the day. These adaptations show how flexible and inventive evolution can be, reshaping a universal process to suit different environments. Yet the essence remains. Light is captured, carbon is fixed, sugars are formed, oxygen is released. It is humbling to consider that the oxygen filling your lungs tonight was likely created in a leaf, a blade of grass, a patch of algae drifting in the sea. Photosynthesis in the oceans, carried out by tiny organisms called phytoplankton, produces much of the Earth's oxygen, sustaining marine life and contributing to the air we breathe. Forests, too, play their role. Vast canopies of leaves lifting toward the sun, each one a small solar collector, quietly converting light into life. On a molecular level, photosynthesis is a balance of precision and abundance. Every photon is captured with care, every electron passed along a chain, every proton gradient built and used. Yet it happens on such a vast scale that its effects shape the entire planet. The balance of gases in the atmosphere, the flow of carbon through ecosystems, the growth of forests and crops, all are tied to this quiet chemistry. As you reflect on photosynthesis, you may see it as a cycle paired with respiration. Plants release oxygen, which animals and humans inhale. Animals release carbon dioxide, which plants draw in. Energy flows from sunlight to sugar, from sugar to ATP, from ATP to work, and back again in continuous exchange. It is not two separate processes, but one great circle. Life breathing in and out, plants and animals intertwined. Tonight, as you rest, this cycle continues beyond your awareness. Leaves turn toward the last traces of sunlight. Algae float beneath the surface of the sea. Chloroplasts hum with quiet activity. The sugars formed will travel through veins and stems, feeding growth, storing energy, and sharing their bounty with the world. The oxygen released will join the atmosphere, diffuse into your lungs, and fuel your own respiration. You are part of this exchange, not separate from it, but woven into it, a participant in the great dialogue between light and life. And so, photosynthesis is more than a process. It is the source of abundance, the quiet alchemy that turns light into matter, starlight into sweetness, photons into breath. As you drift into sleep, you may picture fields of green stretching beneath the sun, forests whispering in the breeze, oceans glimmering with life, all engaged in this silent work. In every moment of daylight, photosynthesis unfolds, and in every moment of night, its products sustain the living world. It is a reminder that life is deeply connected to light, that the chemistry of life begins with the sun, and that even in stillness, the legacy of sunlight flows through you. 14. Metabolic pathways. When we think of metabolism, it is easy to imagine a single process, a fire burning within us that consumes food and releases energy. Yet metabolism is not a single flame, but a vast network of pathways, weaving together like rivers across a landscape. Each pathway is made of many steps, each step guided by enzymes, each enzyme tuned to a particular transformation. Together, they create a harmony where molecules flow, combine, separate, and change, always with purpose, always in balance. Metabolism is the quiet choreography of life, a dance of molecules that continues in every cell, unceasing, precise, and resilient. At the most basic level, metabolism can be divided into two great streams, catabolism and anabolism. Catabolism is the breaking down of larger molecules into smaller ones, releasing energy in the process. It is the dismantling of glucose during respiration, the splitting of fatty acids, the deconstruction of proteins. Anabolism, in contrast, is the building up of larger molecules from smaller ones, requiring energy to do so. It is the construction of proteins from amino acids, the weaving of DNA from nucleotides, the assembly of membranes from lipids. One provides energy, the other consumes it. One releases, the other gathers. Together, they form a cycle that sustains the flow of life. Within this cycle are countless pathways, many of them interconnected. Consider glycolysis, which breaks down glucose into pyruvate. Pyruvate can then flow in several directions. It may enter the citric acid cycle, producing ATP. It may be converted into lactate during fermentation, or into other precursors for biosynthesis. In this way, a single molecule serves as a crossroads, and enzymes act as gatekeepers, directing it where it is most needed. Such crossroads exist throughout metabolism, where molecules can follow one of several paths, ensuring flexibility and adaptation to changing conditions. Central to many pathways is the concept of energy carriers. ATP, NADH, FADH, and NADPH are molecules that hold energy or electrons temporarily, passing them from one reaction to another. These carriers are like couriers moving through a city, delivering their cargo to where it is required. Without them, energy would be lost, scattered, or wasted. With them, it is delivered precisely, in the right amount, at the right time. The interplay of carriers ensures that catabolic and anabolic pathways are linked. that energy released in one place can be used in another. The elegance of metabolic pathways lies not only in their complexity but in their regulation. Cells cannot afford to let every reaction run unchecked. Instead, enzymes are regulated through feedback, signals, and modifications. If a product accumulates beyond need, it gently slows the pathway that produces it. If energy is scarce, pathways accelerate to produce more ATP. Hormones, too, influence metabolism, carrying signals between tissues. Insulin encourages cells to take up glucose and build reserves, while glucagon signals the release of stored energy. Adrenaline, cortisol, and other messengers add further layers of control, ensuring that the needs of the whole body are met. One striking feature of metabolism is its conservation across life. The glycolytic pathway, the citric acid cycle, and the electron transport chain are nearly universal, found in bacteria, plants, animals, and fungi. The enzymes may vary slightly, the regulation may differ, but the core steps are the same. This universality speaks to the ancient origins of metabolism, shaped billions of years ago and preserved because of its efficiency and resilience. To study these pathways is to glimpse the shared heritage of all living beings, a chemical language written into the fabric of life itself. Metabolism also reveals the interconnectedness of nutrients. Carbohydrates, lipids, and proteins do not flow through separate channels but intersect and overlap. Excess glucose can be stored as glycogen or converted into fat. Amino acids can be burned for energy or used to build proteins. Fatty acids can be broken down into acetyl-CoA, feeding into the citric acid cycle alongside glucose. Nothing is wasted. Everything can be transformed. This flexibility allows life to endure scarcity, abundance, or change, always finding a way to keep the flow of energy steady. In plants, metabolism takes on additional layers. Photosynthesis produces sugars, which can be used immediately for energy or stored as starch for later. These sugars feed into the same glycolysis and citric acid cycle as in animals, highlighting again the universality of life's chemistry. Plants also synthesize unique compounds, pigments, toxins, aromas, specialized molecules that serve ecological roles. Some deter herbivores, others attract pollinators, others protect against pathogens. These secondary metabolites are reminders that metabolism is not only about survival, but about interaction, adaptation, and connection to the wider environment. On a planetary scale, metabolism shapes ecosystems. The carbon fixed by photosynthesis flows into herbivores, predators, and decomposers. Nitrogen, essential for amino acids and nucleotides, cycles through soil, plants, animals, and microbes. Oxygen and carbon dioxide ebb and flow through respiration and photosynthesis, balancing the air we breathe. Metabolism is not confined to cells but expands outward, weaving together organisms, communities, and the biosphere. Every living thing participates in this network, drawing from it and contributing to it, sustained by the same cycles of transformation. In humans, metabolism also adapts to rhythms, the daily cycle of waking and sleeping, the longer cycles of feeding and fasting. During the day, glucose from food provides immediate fuel. At night, glycogen stores are tapped, and fat reserves contribute more. In fasting, the body shifts to rely on fat and produces ketone bodies as alternative fuels for the brain. These shifts illustrate the flexibility of metabolism, its ability to adjust smoothly to sustain life in different circumstances without interruption. As you drift deeper into stillness, you may imagine metabolism as a vast web within you, threads of reactions crossing and joining, energy flowing along the strands. At one point, glucose is broken apart. At another, ATP is formed. Elsewhere, proteins are built, lipids are stored, nucleotides are assembled. The web vibrates with activity, yet it is orderly, not chaotic. Each step is guided, each pathway regulated, each molecule part of a larger design. And in this web lies a quiet lesson. Life is not sustained by one process alone, but by countless processes connected together. Metabolism is a reminder of interdependence, of balance, of continuity. Just as ecosystems rely on the interplay of many species, cells rely on the interplay of many pathways. Each is small, yet together they create the flow of life. So, as you rest, metabolism continues within you. Molecules move, enzymes act, carriers deliver, pathways shift, energy is balanced, matter is renewed, order is maintained. It is a dance that began in the earliest cells and continues tonight within you, unbroken, patient, and enduring. 15. The flow of information. From DNA to protein. Among the many wonders of biochemistry, few are as elegant as the way information flows within living systems. Life depends not only on energy and structure, but also on instructions, the careful transfer of knowledge from one molecule to another. This flow of information, from DNA to RNA to protein, is often called the central dogma of biology. Though the name may sound rigid, in truth it is a gentle sequence, a choreography that allows the blueprint of life to become the machinery and form of the living being. It begins with DNA, the archive of information, resting in the nucleus of eukaryotic cells or in the cytoplasm of simpler organisms. DNA contains the recipes for proteins, but the recipes must first be copied into a form that can leave the archive. This step is transcription, the writing of RNA from a DNA template. Within the nucleus, enzymes unwind a section of DNA, exposing its bases. RNA polymerase, the key enzyme, reads one strand of DNA and builds a complementary strand of RNA. If the DNA contains an adenine, the RNA polymerase adds a uracil in place of thymine. If it contains a guanine, the enzyme adds a cytosine. Step by step, nucleotide by nucleotide, a messenger RNA molecule is formed. Messenger RNA, or mRNA, is like a photocopy of a page from a book. The full genome is too vast to leave the archive, but mRNA carries the instructions for a single protein. Once complete, the mRNA leaves the nucleus through small pores and enters the cytoplasm, where it will be read and translated. The original DNA remains safe, undisturbed in its chromosomal coils, while the temporary RNA carries its message forward. The process of translation begins at the ribosome, a complex molecular machine made of both protein and ribosomal RNA. The ribosome reads the mRNA in groups of 3 bases, called codons. Each codon corresponds to one amino acid, the building blocks of proteins. Transfer RNA molecules, or tRNAs, bring amino acids to the ribosome. Each tRNA has an anticodon that pairs with the codon on the mRNA, ensuring accuracy. When a match is made, the ribosome links the amino acid to the growing chain, and the process continues, codon by codon, until the protein is complete. This translation is both precise and flexible. The genetic code, made of 64 possible codons, covers all 20 amino acids, with some codons serving as start and stop signals. Remarkably, this code is nearly universal across all life. The same codons specify the same amino acids in bacteria, plants, and humans. This universality speaks to a shared origin, a common language that has endured for billions of years. It is a thread connecting all living beings, a reminder that we are participants in one long story written in nucleotides and proteins. Once a protein is synthesized, it often requires folding and modification to become fully functional. The sequence of amino acids dictates how the chain folds into helices, sheets, and coils, creating the three-dimensional shape needed for activity. Molecular chaperones assist in this process, preventing misfolding. In some cases, proteins are also modified after translation. Sugars or lipids may be attached, phosphate groups may be added, or segments may be cleaved away. These modifications fine-tune the protein, preparing it for its specific role, whether as an enzyme, a transporter, a receptor, or a structural component. The flow of information does not stop with translation. Proteins themselves influence the activity of DNA, turning genes on or off, amplifying signals, or silencing unnecessary instructions. Feedback loops ensure balance. If too much of a protein is produced, the cell reduces transcription. If more is needed, transcription is increased. This regulation ensures efficiency, preventing waste while adapting to change. The process is not rigid but responsive, adjusting continuously to the needs of the cell and the organism. It is striking to realize that the traits of an organism, from the color of a flower to the texture of hair, arise from this simple flow. DNA to RNA to protein. Each protein plays its part, and together they create the complexity of life. The process is not chaotic but ordered, not haphazard but guided, and though it is intricate, it is also deeply reliable. Billions of times each day in each cell of your body, DNA is transcribed, RNA is translated, proteins are formed, and yet mistakes are rare, caught by proofreading mechanisms and corrected by repair systems. The fidelity of this flow is what allows life to endure. In multicellular organisms, this flow is coordinated across countless cells. In one cell, certain genes are active, producing proteins that define its role as a muscle cell, a neuron, or a skin cell. In another, different genes are active, creating a different role, yet the DNA is the same in all. The difference lies in which instructions are expressed, which pages of the genetic book are read, and which remain closed. This selective expression allows specialization, creating tissues, organs, and systems from the same underlying instructions. On a larger scale, the flow of information connects generations. DNA is passed from parent to offspring, carrying the instructions for life. The same process of transcription and translation continues, ensuring that the traits encoded in the DNA are expressed in the new individual. Mutations may introduce variations, leading to diversity and evolution, but the flow itself remains constant. From the earliest cells to the present, the path of DNA to RNA to protein has guided life, unbroken, faithful, and enduring. As you rest, this flow continues within you. In your brain, neurons express proteins that allow them to send signals. In your muscles, proteins contract to maintain posture. In your skin, proteins form barriers against the outside world. In your blood, proteins carry oxygen, defend against pathogens, and repair wounds. Each one began as a sequence in DNA, transcribed into RNA, translated into protein, folded into shape, and set to work. You need not direct it. It happens silently, invisibly, faithfully. And so, the flow of information is more than a molecular process. It is the quiet story of how life's instructions are read and carried out, how the blueprint becomes reality, how potential becomes action. It is the gentle conversion of sequence into structure, of letters into form, of information into living function. Tonight, as you drift into deeper stillness, this flow continues in every cell of your body, a soft current of transcription and translation, writing and reading, shaping the chemistry of life. 16. Regulation and control in biochemistry. The chemistry of life is not only about building molecules, breaking them down, and transferring energy. It is also about balance. Within each cell, countless reactions are occurring at once, each with its own needs, timing, and requirements. Without coordination, these reactions would collide in confusion, wasting resources, producing too much of some molecules and too little of others. Yet life is not chaotic. It is ordered, responsive, and adaptable. This order arises through regulation, the gentle control of biochemical processes to maintain harmony. Regulation ensures that cells produce what they need, when they need it, and in the right amounts, neither more nor less. At the heart of this regulation are enzymes, the catalysts that guide reactions. Enzymes do not work blindly. Their activity is finely tuned by feedback from the molecules they help create. If a product begins to accumulate, it may bind to the enzyme that started the pathway, gently slowing it down. This is known as feedback inhibition, a common form of control. It is as if the pathway listens to its own output, adjusting itself to prevent excess. When the product is used up, the inhibition eases, and the pathway resumes. In this way, cells maintain balance automatically, without waste or shortage. Another form of regulation comes through allosteric control. Many enzymes have sites beyond their active site, places where small molecules can bind and change their shape. When a molecule binds at this allosteric site, the enzyme may become more active or less active, depending on the signal. This allows the enzyme to respond not only to its own products but also to signals from other pathways, creating a web of communication within metabolism. Beyond individual enzymes, regulation also occurs at the level of gene expression. A cell does not need to produce every protein at all times. Instead, it controls which genes are transcribed into RNA, and therefore which proteins are made. Some genes are always active, producing essential proteins needed for survival. Others are turned on only in certain conditions, such as when nutrients are available or when stress occurs. Transcription factors, proteins that bind to DNA, act as switches, controlling whether genes are read or left silent. In this way, the cell tailors its functions to its circumstances, conserving energy and resources. In multicellular organisms, regulation extends further, guided by hormones and signals that coordinate the activity of entire tissues and organs. Hormones are chemical messengers released into the bloodstream, carrying instructions from one part of the body to another. Insulin, for example, signals cells to take in glucose after a meal, storing it as glycogen or fat. Glucagon, in contrast, signals the release of glucose when blood levels fall. These two hormones act in balance, keeping blood sugar within a narrow range. Other hormones regulate growth, reproduction, metabolism, and stress responses, weaving the activity of many organs into a single coordinated whole. Signals also flow through the nervous system, where neurotransmitters pass between neurons, triggering cascades of biochemical reactions. These signals can cause rapid changes in enzyme activity, altering metabolism in seconds. The body thus has both long-term regulation through hormones and immediate regulation through neural signals, a dual system that ensures both stability and responsiveness. Another important form of regulation is compartmentalization. Cells are not homogeneous. They are divided into organelles, each with its own environment. Enzymes and pathways are separated into these compartments, preventing interference and allowing specialized conditions. The breakdown of glucose, for example, begins in the cytoplasm but continues in the mitochondria. DNA transcription occurs in the nucleus, while protein synthesis happens in the cytoplasm. This spatial organization is itself a form of control, guiding molecules along particular paths and ensuring order. Regulation also occurs through cycles of modification. Proteins may be activated or inactivated by the addition of phosphate groups, a process called phosphorylation. Enzymes, called kinases, add these groups, while phosphatases remove them. This reversible modification acts as a switch, turning proteins on or off as needed. Other modifications, such as acetylation, methylation, or ubiquitination, provide additional layers of control, marking proteins for activity, silence, or even destruction. At the molecular level, regulation creates rhythms. In some cases, these rhythms are tightly timed, as in the oscillations of enzymes that control the cell cycle, ensuring that cells divide only at the right moment. In others, the rhythms are daily, tied to the circadian clock, which aligns metabolism with the cycle of light and dark. These rhythms allow organisms to anticipate changes in their environment and prepare accordingly, saving energy and enhancing survival. On a broader scale, regulation is not only about molecules but about the balance of the organism with its environment. When food is abundant, metabolism shifts to store energy. When food is scarce, metabolism shifts to conserve and mobilize reserves. When temperature drops, metabolism adjusts to generate heat. When stress occurs, hormones prepare the body for action. These adjustments happen seamlessly, without conscious effort, guided by signals that coordinate countless cells into unified responses. It is remarkable to reflect that this regulation is achieved not through a central command, but through distributed control. Each enzyme, pathway, and cell responds to local signals, and together, their interactions create balance. It is like a murmuration of starlings, where each bird follows simple rules, yet the flock moves in graceful harmony. So too, with biochemistry, local interactions give rise to global order. As you rest, this regulation is occurring within you. Your blood sugar is being monitored, your enzymes are adjusting their pace, your hormones are circulating, your genes are being switched on and off. You do not feel these processes, yet they maintain your health, your stability, your continuity. They ensure that you have the right amount of energy, that your cells produce the proteins they need, that your body responds gently to change. And so, regulation in biochemistry is more than control. It is balance, harmony, adaptation. It is the way life sustains itself, not through rigidity, but through flexibility, not through command, but through cooperation. Tonight, As you drift deeper into stillness, these processes continue within you, silent and precise, maintaining the gentle equilibrium that sustains your life. 17. The chemistry of cell membranes. Every living cell is surrounded by a boundary, a delicate yet resilient structure known as the cell membrane. It is not a wall of stone or steel, not an impenetrable barrier, but a soft, flexible sheet made of molecules. Its purpose is not only to enclose but to protect, not only to separate but also to allow exchange. Without membranes, life would dissolve into its surroundings, molecules drifting without order, reactions losing their direction. With membranes, cells have form, integrity, and the ability to control their inner world. The chemistry of membranes is thus the chemistry of boundaries, of selective passage, of gentle control at the interface between inside and outside. The fundamental structure of a cell membrane is the lipid bilayer, a double layer of molecules called phospholipids. Each phospholipid has two parts, a head that is attracted to water and two tails that are repelled by it. When placed in water, phospholipids arrange themselves so that their heads face outward toward the water and their tails cluster inward, away from it. This spontaneous arrangement creates a bilayer, with a watery environment on both sides and a protected, oily interior between the layers of tails. This bilayer is stable yet flexible, forming the foundation of every cell membrane. The bilayer alone would be too simple to support the needs of life. Into it are inserted proteins, cholesterol, and other molecules that add function and complexity. Membrane proteins serve as channels, transporters, receptors, and anchors. Some span the entire bilayer, creating passageways for ions or nutrients. Others sit on the surface, acting as sensors that detect signals from outside the cell. Cholesterol, interspersed among the phospholipids, adds stability, preventing the membrane from becoming too rigid in the cold or too fluid in the heat. Together, lipids, proteins, and cholesterol form what is called the fluid mosaic model, a dynamic, shifting arrangement where components move freely within the plane of the membrane, like boats drifting on a gentle sea. One of the most essential roles of the membrane is selective permeability. It allows certain molecules to pass while excluding others. Small, uncharged molecules such as oxygen and carbon dioxide can slip through easily, diffusing across the bilayer. Water also passes, though more slowly, often with the help of specialized channels called aquaporins. Larger molecules, or charged ions, cannot cross freely and require transport proteins. Some transport is passive, flowing with concentration gradients, while others are active, requiring energy, often in the form of ATP, to move molecules against the gradient. In this way, the membrane maintains differences between the inside and outside, creating conditions necessary for life. These differences are not random. They are carefully maintained. Inside the cell, potassium ions are more abundant, while outside, sodium ions dominate. This imbalance is upheld by the sodium-potassium pump, a membrane protein that uses ATP to move sodium out and potassium in. The result is an electrochemical gradient, a difference in charge and concentration across the membrane. This gradient stores potential energy, like water behind a dam, ready to be used for processes such as nerve impulses, muscle contraction, and the transport of nutrients. Membranes are also sites of communication. Receptor proteins embedded in the bilayer detect signals such as hormones, neurotransmitters, or nutrients. When a signal binds, the receptor changes shape, setting off cascades of reactions within the cell. This allows cells to respond to their environment, to grow when conditions are favorable, to defend when threats are present, or to adjust metabolism to new circumstances. In multicellular organisms, membrane communication coordinates tissues and organs, ensuring that the body functions as a whole rather than as isolated parts. Beyond communication, membranes also provide identity. Attached to the outer surface of many membrane proteins and lipids are chains of sugars, forming structures known as glycoproteins and glycolipids. These act as molecular signatures, distinguishing one cell from another. In the immune system, for example, such signatures allow the body to recognize its own cells and distinguish them from invaders. In development, they help cells organize into tissues, guiding them to their correct positions. The sugars are like labels written in a molecular script, silently informing each cell of its place in the larger organism. Inside the cell, membranes do not stop at the boundary. Organelles, such as the nucleus, mitochondria, endoplasmic reticulum, and Golgi apparatus, are all enclosed by membranes. These internal membranes create compartments, each with its own conditions and tasks. The nucleus, wrapped in a double membrane, protects the DNA. The endoplasmic reticulum, a labyrinth of membranes, provides sites for protein and lipid synthesis. The Golgi apparatus sorts and packages molecules for transport. Mitochondria and chloroplasts, with their folded membranes, generate energy. In this way, membranes divide labor within the cell, allowing complex tasks to be performed efficiently and without interference. The flexibility of membranes also allows them to change shape. Cells can engulf large particles or liquids through endocytosis, folding the membrane inward to form vesicles that carry material inside. Conversely, cells can release substances through exocytosis, fusing vesicles with the membrane to expel their contents. These processes allow cells to import nutrients, expel waste, communicate with neighbors, and reshape their surroundings. The membrane is not a fixed boundary but a living, moving interface, adapting constantly to the needs of the cell. On a larger scale, membranes contribute to movement and sensation. In nerve cells, rapid changes in membrane potential create action potentials, the signals that carry thoughts and sensations. In muscle cells, these signals trigger contractions, allowing movement. In sensory cells, membranes contain proteins that respond to light, sound, touch, or chemicals, converting external stimuli into signals the body can interpret. In each case, the membrane is more than a boundary. It is the stage upon which communication, sensation, and motion are possible. Despite their importance, membranes are fragile. Heat, detergents, or certain toxins can disrupt the bilayer, dissolving its structure. Yet in living systems, membranes are constantly repaired, lipids replaced, proteins recycled, structures renewed. Their resilience comes not from being unbreakable but from being dynamic, always in motion, always maintained. It is this dynamism that allows cells to endure, even in the face of stress. As you reflect on the chemistry of membranes, you may picture them not as rigid walls, but as shimmering, flexible films, alive with motion. Lipids drift, proteins shift, sugars wave gently on the surface, molecules pass through channels, signals ripple across receptors. Each membrane is a living interface, connecting inside and outside, order and environment, stability, and change. And so, cell membranes remind us of the importance of boundaries that are not barriers but gateways, boundaries that protect yet also connect. They are the reason cells can maintain their inner worlds, yet also the reason they can sense and respond to the outer world. They embody the balance of separation and connection, the balance that sustains life. Tonight, as you rest, the membranes of your cells are at work. Nutrients enter, wastes leave, signals are received, impulses are sent. Your heart beats because of ion flows across membranes. Your thoughts travel because of potentials generated by membranes. Your senses respond because of proteins embedded in membranes. All of this continues without your effort, without your awareness, sustained by the quiet chemistry of the bilayer. 18. Molecular communication and signaling pathways. Life does not happen in isolation. Even the smallest cell, floating alone in water, is constantly receiving and responding to signals. Within the body, where trillions of cells coexist, communication is essential. Cells must know when to divide, when to grow, when to conserve energy, when to defend, and when to rest. Without communication, the body would fall into disarray. Organs would lose coordination, and tissues would drift without purpose. Yet with communication, the many cells of the body act as one, as if guided by a shared rhythm. This quiet guidance comes through molecular signaling, the language of biochemistry that allows cells to speak to one another. Communication begins when one cell releases a signal. This signal may be a small molecule, a peptide, a lipid, or even a gas. It travels across space, sometimes only to a neighboring cell, sometimes into the bloodstream to reach distant tissues. The receiving cell must then detect the signal, which it does through receptors. These receptors are proteins, often embedded in the cell membrane, that can recognize and bind to specific signals. The binding is precise, like a key fitting into a lock. Once bound, the receptor changes shape, and this change sets off a cascade of reactions within the cell. These cascades are known as signaling pathways. A single signal at the surface is amplified inside, passing from molecule to molecule, often through phosphorylation or other modifications. Each step acts as a relay, spreading the message, refining it, and directing it to the appropriate targets. In this way, a tiny concentration of hormone or neurotransmitter outside the cell can lead to large, coordinated changes inside. The cell may alter gene expression, change its metabolism, adjust its movement, or prepare for division. There are many types of signaling, each suited to a different context. Autocrine signaling occurs when a cell releases signals that act on itself, allowing self-regulation. Paracrine signaling occurs between nearby cells, important for local coordination. Endocrine signaling uses hormones carried by the blood, reaching distant organs. Synaptic signaling, unique to neurons, transmits messages rapidly across tiny gaps called synapses. Each type shows the adaptability of communication, allowing signals to travel near or far, quickly or slowly, broadly or precisely. One well-studied example is the signaling of insulin. After a meal, glucose levels rise in the blood. The pancreas releases insulin, which travels through the bloodstream to many tissues. Cells with insulin receptors respond by taking in glucose, storing it, or using it for energy. At the molecular level, insulin binding activates a receptor kinase, which phosphorylates proteins inside the cell. setting off a cascade that ultimately moves glucose transporters to the membrane. The result is the lowering of blood sugar, restoring balance. This pathway shows how a single signal can coordinate activity across the entire body, linking one organ to many others. Another example is the signaling of adrenaline, also known as epinephrine. In moments of stress, the adrenal glands release adrenaline into the blood. It binds to receptors on heart cells, causing them to beat faster, on liver cells, causing them to release glucose, on fat cells, causing them to release fatty acids. All of these responses prepare the body for action, even though they occur in different tissues. The key lies in the receptor and the pathways it activates. Each cell interprets the same signal in a way suited to its role, creating a coordinated response. Signaling pathways often intersect and influence one another, forming networks rather than simple lines. A signal may activate one pathway that promotes growth while inhibiting another that promotes rest. Another signal may do the opposite. The balance between these pathways determines the final outcome. In this way, cells integrate multiple messages, weighing them to decide their course of action. This integration allows for subtlety, preventing overreaction and enabling precise control. Within the cell, second messengers play an important role in spreading signals. These are small molecules such as cyclic AMP, calcium ions, or inositol triphosphate. When a receptor is activated, it may trigger the production or release of these messengers. Because they can diffuse rapidly inside the cell, they carry the signal far and wide, activating many targets at once. In neurons, for example, calcium ions flood in during signaling, triggering neurotransmitter release and activating proteins involved in memory and plasticity. Second messengers are thus essential amplifiers, ensuring that a small signal at the surface creates a meaningful change within. Communication is not only about stimulation but also about restraint. Just as pathways can be activated, they can also be silenced. Phosphatases remove phosphate groups, reversing phosphorylation. Enzymes break down second messengers, stopping their diffusion. Receptors themselves may be internalized or degraded when overstimulated. These mechanisms ensure that signals do not persist longer than needed, preventing chaos. The body is not designed for constant activation, but for balance, with signals rising and falling like waves. On a larger scale, signaling underlies development, guiding cells to form tissues and organs. During embryonic growth, gradients of signaling molecules tell cells where they are, what type of tissue to become, and when to divide. A cell at one end of the gradient may become part of the nervous system, while a cell at the other end becomes part of the skin. This orchestration builds complexity from simplicity, structure from a single fertilized cell. Even in adulthood, signaling continues to guide repair, regeneration, and adaptation. Cellular communication also governs the immune system, where signaling molecules called cytokines coordinate defense. When a pathogen is detected, immune cells release cytokines that recruit other cells, stimulate inflammation, and increase activity. When the threat is over, other signals calm the response, preventing damage to the body's own tissues. This interplay of signals allows the immune system to be both powerful and precise, defending without overwhelming. At the smallest scale, even bacteria communicate. Through a process called quorum sensing, bacteria release small molecules into their environment. As the population grows, the concentration of these molecules rises. When a threshold is reached, the bacteria sense it and change their behavior together, perhaps forming a biofilm or producing light. This shows that signaling is not limited to complex organisms but is a universal feature of life. As you rest, countless signals are flowing within you. Hormones move through your blood, carrying instructions from one organ to another. Neurotransmitters pass across synapses, guiding thoughts and dreams. Signals at the surface of your cells regulate metabolism, growth, and repair. These pathways ensure that your body remains coordinated, that your cells act not as isolated units but as parts of a greater whole. And so, molecular communication is more than chemistry. It is the language of life, a dialogue written not in words but in molecules, a conversation that has continued for billions of years. It is the reason your heart beats in rhythm, your thoughts form, your body heals. Tonight, as you drift deeper into sleep, these signals continue their quiet work, carrying messages through your cells, weaving them into harmony. 19. The chemistry of the genetic code. At the very core of life's continuity lies a code, a language written not in letters or sounds but in molecules. This is the genetic code, the quiet set of rules by which the sequences of nucleotides in DNA and RNA are translated into the sequences of amino acids in proteins. Though it appears simple, with just 4 nucleotides forming triplets, the genetic code is universal, profound, and enduring. It is the bridge between information and form, between the archive of DNA and the living machinery of proteins. To reflect upon it is to glimpse the underlying unity of all life, the common thread that ties together the simplest microbe and the most complex organism. The code is built from nucleotides, the basic units of DNA and RNA. In DNA, these are adenine, thymine, cytosine, and guanine. In RNA, thymine is replaced by uracil. When arranged in long chains, these nucleotides form sequences that carry instructions. Yet the instructions must be read in groups of 3, known as codons. Each codon specifies one amino acid, the building block of proteins. With 4 nucleotides arranged in triplets, there are 64 possible codons, enough to encode the 20 amino acids found in proteins, along with start and stop signals that define the boundaries of each sequence. This triplet system is both efficient and elegant. Because there are more codons than amino acids, the code is redundant, meaning that several codons can specify the same amino acid. This redundancy provides a measure of protection against mutations. A change in the third position of a codon often does not alter the amino acid it encodes, allowing proteins to be built correctly even in the presence of small errors. This feature, sometimes called wobble, shows the code's resilience, its capacity to preserve meaning despite variation. The universality of the code is perhaps its most striking feature. In nearly every organism, from bacteria to humans, the same codons specify the same amino acids. The codon AUG begins translation, marking the amino acid methionine as the starting point. Codons UAA, UAG, and UGA signal the end, instructing the ribosome to stop. This consistency means that the genetic code was established very early in the history of life and has been conserved for billions of years. It is a shared language, spoken across all domains of life, a reminder that all organisms are connected by descent from common ancestors. To translate the code, cells rely on transfer RNA, or tRNA. Each tRNA carries an amino acid and has an anticodon region that pairs with a codon on messenger RNA. This pairing is precise. ensuring that the correct amino acid is added to the growing protein chain. The ribosome, acting as both guide and catalyst, moves along the mRNA, reading codons in sequence, while tRNAs bring amino acids one by one. In this way, the abstract information of nucleotides is transformed into the tangible form of a protein, a molecule that can fold, act, and contribute to the life of the cell. The code is not merely functional, it is foundational. It allows DNA to serve as a long-term archive of information, RNA to act as a messenger and intermediary, and proteins to serve as the machinery and structure of life. Without the code, the flow of information would halt. With it, life can not only exist but evolve, adapting and diversifying while retaining continuity. There are rare exceptions to the universality of the code. In some mitochondria and in a few single-celled organisms, certain codons are reassigned, specifying different amino acids than in the standard code. Yet these variations are minor, more like dialects of a shared language than separate tongues. The overwhelming consistency of the code across life underscores its stability and success, its ability to endure and sustain. The genetic code also provides the foundation for heredity. Mutations in DNA alter codons, which may alter amino acids in proteins. Some mutations are silent, changing a codon without changing the amino acid. Others are conservative, substituting one amino acid for another with similar properties. Still others have profound effects, altering protein shape and function. Through these changes, new traits arise, some beneficial, some neutral, some harmful. Over generations, this variation fuels evolution, the gradual shaping of life's diversity. The code, stable yet flexible, provides both continuity and change. On a planetary scale, the genetic code unites all living beings. The same codons that guide protein synthesis in a bacterium guide it in an oak tree, a dolphin, or a human. The same triplets that begin and end genes in a microbe do so in your own cells. This shared foundation allows scientists to read the DNA of any organism, to compare sequences across species, to trace relationships through time. It allows genes from one organism to be expressed in another, enabling biotechnology to create medicines, crops, and therapies. The code is not locked within species but is universal, open, and readable by all. To imagine the genetic code is to imagine a vast text, written in 4 letters, read 3 at a time. It is a text without punctuation, yet it is understood with clarity. It is a language without sound, yet it speaks across billions of years. Each triplet is small, yet together they form the instructions for life itself. And though the code is molecular, its implications are cosmic, connecting every form of life on Earth in one continuous story. As you rest, this code is being read within you. In your cells, ribosomes trace along mRNA, reading codons, pairing anticodons, joining amino acids. Proteins are being formed in your muscles, your skin, your neurons, your blood. Each protein is a sentence written by the code, folded into shape, carrying out its role. You do not sense it, yet it sustains you. The code is silent, but its effects are everywhere, from your heartbeat to your thoughts. And so, the genetic code is more than chemistry. It is the script of life, the silent language that bridges molecules and meaning, information and action. It is both ancient and immediate, both universal and personal. Tonight, as you drift deeper into sleep, this code continues its quiet work within you. writing the story of life, letter by letter, codon by codon, a story that began long ago and continues still, unbroken. 20. Proteins and their structures. Proteins are often called the workhorses of the cell, and with good reason. They carry out an extraordinary range of tasks, from catalyzing reactions to transporting molecules, from transmitting signals to providing structure. Yet behind their versatility lies a simple principle. Proteins are chains of amino acids folded into three-dimensional shapes. The chemistry of these shapes and the rules that guide their formation allow proteins to perform their countless roles. To reflect upon proteins is to reflect upon the subtle ways in which sequence becomes form and form becomes function. Each protein begins as a sequence, a chain of amino acids linked by peptide bonds. There are 20 different amino acids, each with a unique side chain, or R group. Some are hydrophobic, repelled by water. Others are hydrophilic, attracted to it. Some carry charges, positive or negative. Others are neutral. These differences determine how the chain will fold, how the amino acids will interact with one another, and ultimately, what shape the protein will take. The first level of protein structure is the primary structure, the linear sequence of amino acids. This sequence is dictated by the genetic code, written in DNA, transcribed into RNA, and translated at the ribosome. The sequence alone carries all the information needed for the protein to fold. Yet from this simple string of amino acids, more complex structures emerge. The second level is the secondary structure, formed by local interactions between nearby amino acids. The most common secondary structures are the alpha helix and the beta sheet. In an alpha helix, the chain coils into a spiral, stabilized by hydrogen bonds between backbone atoms. In a beta sheet, strands of the chain run alongside each other, connected by hydrogen bonds to form a sheet-like arrangement. These structures are regular, repeating, and elegant, providing stability and flexibility. The third level is the tertiary structure, the overall three-dimensional shape of a single polypeptide chain. Here, interactions among distant amino acids come into play. Hydrophobic side chains cluster inward, away from water, while hydrophilic and charged groups orient outward. Hydrogen bonds, ionic interactions, van der Waals forces, and disulfide bridges all contribute. The result is a unique shape, folded with precision, capable of specific functions. It is in this shape that the personality of the protein emerges, whether as an enzyme, a receptor, a transporter, or a structural element. The fourth level, quaternary structure, arises when multiple polypeptide chains come together to form a complex. Hemoglobin, for example, is made of 4 subunits, each capable of binding oxygen, but working together to release it cooperatively. Many enzymes and structural proteins function in this way, not as single chains but as assemblies, where the arrangement of parts enhances the whole. Protein structure is not rigid but dynamic. Proteins breathe, flex, and shift, adjusting their shapes as they work. Enzymes, for example, often change shape when binding substrates, a phenomenon called induced fit. Receptors may alter their conformation when bound by a signal, triggering cascades inside the cell. Structural proteins such as actin and tubulin assemble into filaments and disassemble again, shaping and reshaping the cell. This flexibility is part of their power, allowing them to adapt to changing conditions while maintaining their core roles. To assist in folding, cells rely on molecular chaperones. These helper proteins do not dictate the final shape but guide the folding process, preventing misfolding or aggregation. They act quietly, protecting exposed hydrophobic regions, allowing the protein to reach its correct conformation. Without chaperones, many proteins would fail to fold properly, leading to dysfunction. The presence of these guides shows how even in the precision of chemistry, support and assistance are essential. When proteins fold correctly, they enable life. When they misfold, they can cause disease. Misfolded proteins may clump into aggregates, disrupting cellular function. In some cases, such as in Alzheimer's disease or Parkinson's disease, these aggregates accumulate in the brain, leading to damage. Prion diseases, rare but striking, are caused by misfolded proteins that induce other proteins to misfold in turn. These examples reveal the delicate balance of protein folding, its importance, and its vulnerability. Proteins are not only functional but also structural. Collagen, the most abundant protein in the body, forms strong fibers that give strength to skin, tendons, and bones. Keratin forms hair, nails, and feathers. Actin and tubulin form filaments and microtubules, shaping cells and enabling movement. These proteins show that form itself can be a function, that strength, flexibility, and resilience can be encoded in molecular design. Some proteins act as carriers. Hemoglobin transports oxygen in the blood, myoglobin stores it in muscles, albumin carries fatty acids, and transferrin carries iron. Others act as messengers, such as hormones and receptors, guiding communication within the body. Still others act as catalysts, enzymes that accelerate reactions by factors of millions, enabling life to occur at the pace we know. In each case, the ability of the protein depends on its structure, its folded shape, and the placement of its amino acids. The diversity of proteins is staggering. With 20 amino acids and sequences that can be hundreds or thousands of residues long, the number of possible combinations is astronomical. Yet life uses only a fraction of these possibilities, shaped by evolution into the proteins that sustain it. Each protein is unique, yet all follow the same rules of chemistry, folding, and interaction. It is this shared foundation that allows us to understand them, to predict their behavior, and even to design new proteins for medicine, industry, and research. Proteins also show the adaptability of evolution. Families of proteins share similar structures but have diverged to perform different tasks. Hemoglobin and myoglobin, for example, share a common ancestor but serve distinct roles in oxygen transport and storage. Enzymes may evolve new specificities, receptors, new ligands, structural proteins, new arrangements. This adaptability reveals how small changes in sequence can lead to new functions, expanding the possibilities of life. As you reflect on proteins, you may imagine them as delicate sculptures, each folded into a precise form, each shape reflecting its role. Some are small and compact, others long and fibrous, others massive complexes with many subunits. Together, they form the machinery of life, dynamic yet reliable, intricate yet universal. And as you rest, proteins within you are folding, unfolding, and refolding. Enzymes are catalyzing reactions. Hemoglobin is delivering oxygen. Collagen is supporting your tissues. Receptors are sensing signals. All of this occurs silently, without your awareness. guided by the chemistry of amino acids and the elegance of folding. So, proteins are more than chains of molecules. They are the embodiment of the genetic code, the translation of sequence into structure, the transformation of information into action. They are both fragile and resilient, precise and adaptable, singular and universal. Tonight, as you drift into deeper stillness, this quiet chemistry continues within you, the folding of proteins, the sustaining of life, the artistry of structure written in amino acids.